Hi, hello. Hi students, am I audible? Hello, am I audible? Am I audible? Yes, please. Hi guys, welcome to Unacademy NEET English. I'm your biology educator Ambika Sharma. Welcome to our channel. How are you all doing? Hello. How are you all doing? I hope I'm perfectly audible and visible to all of you. Yes, I hope I'm perfectly audible and visible to all of you. Yes. If it is a yes, do let me know in the chat section with some fire emojis. If it is a yes, if I'm perfectly audible and visible to you guys, then do let me know in the chat section with some fire emojis. Welcome to the class, guys. Welcome. I am good, guys. Thank you so much for asking. Welcome to the session. Welcome to the session. Thank you so much. Thank you so much. I hope you guys are enjoying the lectures. I hope you people are completing your syllabus because we have 6 months only, right, bache? And in these 6 months, we have to complete our syllabus. We have to practice the PYQs. We have to give the mock test as well. Just imagine. So we have a lot of work to do. Yes, we have a lot of work to do. So I really want you people to be consistent, to be sincere, follow this series, right, bache? You know that in chemistry, I think 3 to 4 chapters are left if I'm not wrong. Only 3 to 4 chapters are left if I'm not wrong. Yes, and even in biology, we are soon going to finish. I think in the coming week, yeah, biomolecules, plant kingdom, animal kingdom, biological classification, right? And when it comes to the living world and your morphology of flowering plants, yes, we can, for living world and for structural organization in animals, I'm going to put the recorded video because right before the series, right, I completed that chapters. That is why. Okay, that is why. And then you can say that next week we'll finish class 11 syllabus, then we'll move to the class 12 syllabus. So by the end of December we'll finish that as well. Okay, we have to finish it. So maybe I'll start taking 3 marathons a week. Anything is possible, don't worry about this, but all I want is that please be consistent. Please be consistent, guys. Okay, chaliye. For neural control and coordination, yes, we missed that lecture. Good. For that also I have to put the recorded video. Thank you so much for reminding me. Yes, so please be consistent. Okay, so now this particular chapter, that is cell cycle and cell division, right? This particular chapter, that is cell cycle and cell division, it is one of the most important chapters and it is a conceptual chapter. If the concepts are clear to you, trust me, no need to revise it again and again then, right? If you understand, if you people understand, yes, my dear students, if you people understand the concept here, nothing to worry about. Then there is nothing to worry about. You are going to love the chapter and there is no need to revise the things again and again. Okay, chaliye. So let's continue this particular chapter, but before that, let me tell you this thing. Unlock the champion in you. So what's that? The 50% off, right? Before that it was 55% off, but now it is 50% off on all the NEET UG subscriptions, guys, and 1 month free extension is there. Just say, let's say you are buying a course for 10 months or for 12 months, so you will get 1 extra month as well. Isn't it amazing? So in the description box there is a link. Click on that link and be the part. So here they are saying that, yeah, this particular thing, that where you are going to get 50% off, it is valid till 10th of November only. So you can apply the coupon code, you can apply any coupon code, it can be Ambika 10, right? And of the other educators also, we do not have any problem in that. Okay, because we are working together as a team, right? So be the part of these batches, and trust me, it is going to boost your NEET preparation. You know that already, right? The team Avengers is teaching in the Phoenix batches, right? In the Phoenix Masters batch, we are guiding you people, right, bache? So in biology, soon I am going to start class 12th syllabus, right? Right now we are Yeah, we are about to finish cell cycle in our batch. Then we will go for biomolecules, and after that, student, after biomolecules, I'm going to start molecular basis of inheritance in my batch. So everything is connected. Everything is connected, and trust me, if you will be the part of batch, you will understand biology in a better way. After molecular basis of inheritance, we will go for the biotechnology. So I believe if you read these chapters in a sequence, right, you can understand them in a better way. So without any delay, be the part of the batch. In the description box, my dear students, there is a link for this batch, and obviously you can start your preparation. And those who are in class 11, soon moving to class 12, for you people, we have the Nimbus Pro reloaded batch. Nimbus Pro reloaded batch. And here in this batch is just at rupees 5499, right? This batch is also live. So here you can see 500 plus lectures are there, 20 plus tests are there. So in our batches, it is not just the content that you are going to get. You will get the DPPs, you will get the mock test, obviously, right, bache, proper mock test, DPP, question practice, everything will be there, okay? Everything will be there. Chalo. So now let's start the chapter, that is cell cycle and cell division. What's the name of chapter, students? It is cell cycle and cell division. So tell me, what is the importance of this particular phenomena, and what is this cell cycle and cell division? If you know anything related to that, do let me know in the chat section. Yes, if you know anything related to that, do let me know in the chat section. Cell cycle and cell division. Cell cycle and cell division. Yes. Tell me quickly, guys, and quickly hit the like button, share the session link with your friends, tell them I have started the class finally. Finally. Thank you so much. Thank you. So tell me, what do you know about the cell division, and what is the need for a cell to divide? Exactly. So bache, see, when we talk about the cell division, it is something very important. You know that if you are talking about a single cell or of any cell, obviously it has to grow. It will grow, right? It will grow. And in the case of unicellular organism, we will also see that how a cell, when it is growing, it is just dividing into two. But now my point is cell division. What exactly is the cell division? Means cell is dividing, right? A single cell is dividing into two. It is forming the daughter cells. And this is very important. Why it is very important? I'll tell you. I'll give you the example rather than, just say we talk about the zygote. What is zygote? Zygote is a diploid cell. Zygote is a diploid cell, a very important link it is. We know that it is a totipotent cell. We know that it is a totipotent cell, and this cell is having the ability, right? This single cell is having, what is the meaning of being a totipotent cell? Yeah, what is totipotency? What is the meaning of being a totipotent cell? Yeah, what is totipotency? So totipotency, a totipotent cell is the ability of a cell to divide into a complete organism. What's that? It is the ability of a cell to divide into a complete organism. So now, from a single cell, right, a multicellular body is going to arise. Our Chintu will form, right? Our Chintu will form. Are you getting my point? Are you getting my point? Right? Our Chintu will grow. Isn't it from that single cell only? From that single cell only. How is it possible? Obviously, this cell will undergo division. We use the word cleavage for that. Right? So basically, in simple word, I'm saying that yes, if we want to develop into a multicellular body, the single cell has to divide, this zygote has to divide, right? So it will become multicellular, it will divide, one cell will divide into two, two will divide into four, four will divide into, right, 8, and soon a multicellular organism will form. So cell division is something that we need. It is something very important. Chalo, I'll give you one more example. Just say, let's say if you will get a cut, if there is an injury, right, we know that our body is going to heal. Right, we know that our body is going to heal. If there is a cut, if there is an injury, we know that soon it is going to heal. How is it possible? Again, the cell division will occur there. Cell will divide, new cells will grow there, and obviously it is going to recover. So it is something very important. Cell division is something very important. Right, bache? Right, bache? Now, let's take the example of Mr. Amoeba. Even I think in class 9th also. In class 9th also we have discussed that. When you talk about amoeba, a single cell, irregular shaped, right, bache? You know that this single cell is capable of completing all the essential processes which is needed for its survival. In the last chapter, right, on Tuesday, right, you know that we completed the, which chapter? We completed that chapter. What is it? That cell, the basic unit of life. There we discussed it, that a single cell is capable of, it is capable of independent existence. It is having the protoplasm. It is having the genetic material, and that is that protoplasm is the physical basis of life that is required, right? For that, there basically in a single cell there is everything which is required for its survival, right? Even the organelles cannot survive individually, but below the level of cell there is no independent existence, right? But this cell is having everything. The cytoplasm is the main arena of cellular activities. Metabolism will be there, and it will carry out the metabolism, the chemical reaction, and it is going to survive for sure. It is going to survive for sure. Chaliye, now when you are talking about this amoeba, right, this is a single cell. Obviously with time it will also grow. The cytoplasmic growth is also there, right? The cytoplasmic growth is also there. And if you remember, we used to say that single cells, right, single celled organism, they are immortal. You remember this thing? We discussed that single celled organism, unicellular organism, they are immortal, right? They are not going to die ever. Why? Because this single cell, it is growing, it is the one that is dividing into two daughter cells, and this is going to be identical. If you remember binary fission, just when we talk about the fission, binary fission, right? Remember binary fission, one cell is dividing into, right, that cell will, it is a fission, it will break, that single cell will form two daughter cells, if you remember. So obviously that is a form of asexual reproduction, right? That is a form of asexual reproduction, and the daughter cells are identical to the parent cell, right? So we used to say ki yeah, single celled organisms, they are immortal because daughter cells are the clones of the parent cell, right? We know such things. So ultimately, even if it is a sexual reproduction, asexual reproduction, we are talking about the division, right? We are focusing on the division. This division thing is important. The cell has to divide. It is not something which is immortal. For its survival, it has to. We know that. Right, bache? We know that division is something that we discuss, right? We have to discuss this. We know that. Now I'm giving you the example of the single celled organism, and when we talk about the single celled organisms, right, in their case, growth and reproduction, again a living word, the living word chapters concept, right? Growth and reproduction, right? In the case of unicellular organisms, growth and reproduction, they are what? They are mutually inclusive events or exclusive events? Mutually inclusive events or exclusive? You please tell me, what are they? Are they mutually inclusive or exclusive? Tell me. Tell me. People, I know you people are very busy. You have a lot of things to do. Hannah, you have to chit chat or something. But please. Tell me. Mutually inclusive, what is the meaning of that? Just say, let's say if a single cell is growing, right? It is just growing, it is increasing its cytoplasm, it is growing, and what will happen? It will divide into two. So basically it was growing, but otherwise it's like cell is like, yeah, I'm going, and all of a sudden the reproduction occur. Why reproduction? Because the single cell is dividing into two. It is growing, right? So that's why in the unicellular organism we are saying growth and reproduction is mutually inclusive event. You cannot differentiate that. It's kind of same there. They are growing and then they are just dividing into two. We know it very well. But otherwise in multicellular organisms we are not going to see like that, right? But now the point is ki madam, we are talking about the division, why are you mixing the things here? All I'm saying is ki everywhere this division thing is very important, right? So but the question is, why do a cell divide? What is the things that are triggering that division? What is the thing that is saying ki arey yaar, you have to divide? Right, what is this? What is this? So, bache, when we talk about the reasons for the division, that why do a cell divide? So, we talk about the nucleocytoplasmic ratio, which is also known as kern-plasm theory. Nucleocytoplasmic ratio या kern-plasm theory. Okay, this is what we need to talk about. Just for an example, if a cell is small in size, if it is small in size, its nucleus is going to be conspicuous. Even in the last chapter we discussed that, right? Its nucleus is what? Its nucleus is going to be conspicuous. The cytoplasm is going to be dense, right? The nucleus is going to be conspicuous. The cytoplasm is going to be dense, right? Now this nucleus is capable, because cytoplasm is less, this nucleus is conspicuous, it is large, it can take over, right? It can take care the function, right? This nucleus is like, yeah, I can take care the needs of this particular cell, right? Because it is large in size and comparatively the cytoplasm is less. Now after that, when let's say the size cell, because cytoplasmic increase is something continuous, right? In the lifespan of a cell, you will see that cytoplasmic increase will always be there. It's just that ki replication will occur in a particular stage, but cytoplasmic increase is there. So, but the nucleus size here, it will remain same, but now the cytoplasm is more. So when, and this is the large cell. So in that cases, this nucleus is not, it cannot take care of the function of this complete cell, right, bache? So it is kind of, it acts like a signal ki now it's the time for a cell to divide. Now it's the time for a cell to divide. Are you getting my point? So first is kern-plasm theory and nucleocytoplasmic ratio. As per this, division occurs due to disturbance in this, right? Due to disturbance in this nucleocytoplasmic ratio of the cell, right? According to this, division occurs when there is a disturbance in nucleocytoplasmic ratio. Okay, so when there is a disturbance in this nucleocytoplasmic ratio, it's a signal for the division. Are you getting my point, Namita? It is a signal. Signal for what? It is a signal for the division. Is that clear? Is that clear? Yes. Right. So this nucleocytoplasmic ratio या karyoplasmic index of small cell is high. Right. Yes. This kern-plasmic ratio या nucleocytoplasmic index या kern-plasmic ratio of small cell is high. Of small cell is high. So because it is high, right, the kern-plasmic ratio of small cell is high, right, bache? So nucleus can efficiently control the activity of the cell. Are you getting my point? Nucleus can efficiently control the activity of the cell, but that kind of option is not there in the case of large size cell. This is the first reason. Secondly, bache, when your cell is small in size, its surface area is more and its volume is less, right? Because it is having more surface area. More surface area means better exchange will be there, and exchange of the material with surrounding is much needed. Right? You know that it is much needed. It is not a kind of a system where there will be no exchange with the surrounding. Of course not. So basically when a cell is small in size, its surface area is more, its volume is less. Right? So surface area more means better exchange. Quickly the exchange will be there. The cell need will be fulfilled. Now when a cell is large in size, its volume is more, its surface area is less, and that less surface area is not capable of fulfilling the need of the cell. Then it is also a signal that cell has to divide. Is that clear? Is that clear? Right, bache? Right? So surface to volume ratio is also a cause of cell division, right? Yes. surface to volume ratio is also a cause of cell division. Surface to volume ratio. So now here the point is that in a cell, when in a small size cell, surface area is more and volume in comparison to that is less. Surface area is more and volume in comparison to that is less, right? And in large size cell, surface area is less and volume is more. So this surface area is insufficient to draw the material, or you can say that for that exchange of material, right? So this surface area is insufficient. Are you getting my point? This surface area is insufficient to draw the material which is required for the cell activity. So at the such kind of stage also the cell division will occur. So these are the causes of cell division. So if it is clear, do let me know in the chat section. Yes, Vache. If it is clear, do let me know in the chat section. Done? Do let me know in the chat section. Done, Vache? So that is why a cell divide. Okay, so cell division we will study under the cell cycle. Now what is the cell cycle? It is a kind of sequential event, right? This event is taking place in a proper coordinated manner or in a proper sequence, where we will see that cell will show the cytoplasmic growth, where we will see that DNA replication is also there, right, bache? The cell organelle, the cell content will duplicate and equally it will distribute to the daughter cells. So when you are talking about the cell cycle, if I have to define the cell cycle, right, let's first start with that thing first, with a kind of definition there. So here you people can see, and please mark it as well, the sequence of events by which a cell duplicates its genome. The sequence of events by which a cell duplicates its genome plus synthesizes the other constituent of the cell and eventually divide into two daughter cell is termed cell cycle. So what is cell cycle? It is the sequence of events by which a cell is going to duplicate its genome, it will synthesize the other constituents of the cell and eventually it will divide into two daughter cells. It will divide into two daughter cells, right? Now my question here is, do you know what is the meaning of genome? Do you know what is the meaning of genome? जैसे genome माने genetic information, some students define it like, ma'am, genetic information of the cell, right, बच्चे? The chromosomes of the cell, some students define it like this. So when we talk about the genome, we also say haploid content of cell. We also say haploid content of cell. But why? Why we are saying haploid content of cell? Anybody in the class who can tell me? Anybody in the class who can tell me? If not, just wait. I'll explain it. Right, I'll explain it. So now, because we have to cover the NCERT as well, you know that this is the beauty of this particular series, right? This is the beauty of this particular series, because here in this series we are not just, right, completing that chapter, like we are also doing NCERT highlights, right? Chalo. So you know that life will start from single cell, and that single cell is the zygote, the totipotent cell, right, bache? So growth and reproduction, they are the characteristic feature of the cell. They are not the defining properties. They are not the defining properties. They are the characteristic feature of the cell. This point is important. This point is important, that it is the characteristic feature of the cell, right? It is not the defining property. So defining properties which are, you can say that, unique to living organism, and without any exception we will see that in the living organism, right? So that is why it is the characteristic feature, right? So now what are they saying? They are saying that all cells, they reproduce, right, by dividing into two, with each parent cell give rise to two daughter cells. देख रहे हो? Daughter cell is given, not the son cell, not the son cell. जैसे let's say this is a cell, this cell is the parent cell. Now it is dividing. Now these two cells, they are known as daughter cells. So sometimes we all, yeah, you can call them offsprings. We can call them offsprings, and sometimes we also use the word progeny. Progeny matlab jaise let's say this single cell, it is dividing into two. That two cells are further developing into four. That four cells are also further, right? They are also further dividing, right? So can I say that these cells are the progeny of this cell? Can I say that these cells are the progeny of this particular cell A? Right? Matlab they are the basically the like in simple language, if I have to explain it in very simple words, if I have to say, explain it, this is one cell is dividing into two, two cells are dividing into four. So basically it is kind of next generation of that particular cell, progeny, right? Progeny is the word. Okay, bache, what is the word that we have here? Yes, what is the word that we have here? It is the progeny. It is the progeny. Okay, right bache? Right bache? So next generation, yeah, in Hindi you can say santan, right? Further, progeny is the word that we use. Okay bache? Okay, so now, so single cell will divide into two, with each parent cell is giving rise to two daughter cell, right bache? So these newly formed daughter cells can themselves grow, and further they will also divide bache. They will also grow, they will also divide. It's like that. So that is what they are saying, that these newly formed daughter cells can themselves grow and divide, giving rise to a new cell population. Giving rise to a new cell population that is formed by the growth and division of that single parent cell and its progeny. So progeny means that daughter cells, that offsprings, santan. Okay, bache? So in other words, such cycles of growth and division which are allowing that single cell to form a structure having millions of cells. Right, bache? Right? Right, so that is what we are discussing under the cell cycle. So that is how we define the cell cycle. Okay, that is how we define the cell cycle. Okay. Okay, is that clear? So cell division is very important, as I told you. So during division of a cell, DNA replication and cell growth will occur. Cell will grow, cytoplasmic increase will be there, plus the DNA replication will also be there. So cytoplasmic increase will it is going to be there, but when it comes to the DNA replication, it will occur in a specific stage only. Why is it so? Because our body is that having that type of mechanism. Matlab, I still remember I was reading this topic in detail in my masters, and then yeah, during my masters I read this topic in detail, right? There are different proteins that we discuss, right, that are not even in the NCERT, cohesin, condensin, a lot of things are there, right? So it's a very kind of deep topic, and trust me, it's a very lovely topic. So yeah, so when we discuss that, so just we say that in the S phase of the cell cycle, there will be the DNA replication. There is a reason for that. DNA replication cannot start by its own, right? There is a kind of, you can say that some checks which will make sure ki DNA replication will only occur when it is needed. The proteins, there are proteins which are playing the role there. Okay, bache? Chaliye. So cell division is a very important process in all the living organism, right bache? So during the division of cell, DNA replication and cell growth occur. So now what are they saying? They are saying that all these processes, that is your cell division, DNA replication, the division of a cell, the replication of a DNA and cell growth, it take place in a coordinated way, in a proper coordination. It's not like that ki cell is saying I am growing, right, but it is having no idea of DNA replication, right? Yeah, DNA is replicating just randomly and not associated, but the things are in a coordination, right? Jaise, right, there should be a proper coordination, right bache? Jaise our body systems, they work in a coordinated manner, right? Right. And why is it so? For the formation of progeny containing intact genomes. I'll give you one example just to simplify this topic for you. Just ye dekho. In the case of humans, how many chromosomes do we have? We have 46 chromosomes. How many chromosomes do we have? We have 46 chromosomes, or we say we have 23 pairs. We say we have 23 pairs, right? So, matlab 23 chromosomes are coming from mother, 23 chromosomes are coming from father, right? So, even, matlab if there is no defect, you can check my chromosome number, it will be 46. You can check my parents' chromosome number, it is also going to be 46, right? So, generation to generation, if there is no defect, if there is no error, right, then we are going to see 46 chromosomes. right? So 46 chromosomes mane we are talking about the humans, right? And the division and everything is such a way to ensure that the progeny, that the daughter cells, they get that intact genome, they get the chromosome number like this. So if you know, and I believe you know that there is one more division that we study here in this particular chapter, that is meiosis. That is also known as reductional division. That is meiosis, that is also known as reductional division. So it is the one that reduces the chromosome number to half. It is the one that reduces chromosome number to half. Are you getting my point? Reduces the chromosome number to half. Are you getting my point? Are you getting my point? So why this division is required for us? Because it is giving us the haploid. It is giving us the haploid gametes. In the case of males, sperm. In the case of female, ova. So you know that a spermatozoa and ovum both are haploid. They fuses and they result in a diploid zygote. So our zygote is going to be diploid only. So 23 chromosomes from mother, 23 chromosomes from father, and the zygote will be having 46 chromosomes. It will divide and that cells are further having 46 chromosomes. We know it very well, right bache? So because we have the two pairs, right, diploid, basically you are talking about the pairs here, right? Just in our case, because 46 chromosomes are there, so one pair is from mother's side, one set is from mother's side, another is from father's side. So it is making one pair. Are you getting my point? So diploid, that's how we denote it, 2n, and n here is the haploid number of chromosome. Okay? So when I am writing 2n means I am talking about an organism or a cell which is diploid, which is having two sets of chromosome, right bache? So our gametes, they are haploid, H for half. So it is having one set of chromosomes only, right? So diploid organism is having two sets of chromosome and gametes are having one set of chromosome. Is that clear, right, about the diploid and haploid part? Yes. So basically in our body there is a proper system that will ensure that progeny should also have the same chromosome number unless or until a defect is there. So जैसे ये देखो, reductional division is required for the gamete formation, for the haploid cell formation, because during fertilization we know that these cells, they need to fuse, and the zygote that they will form, it will also get 23 chromosome from male, 23 chromosome from female, total 46 chromosomes. Total 46 chromosomes. Is that clear? Is that clear? So although cell growth in terms of cytoplasmic increase, matlab when you talk about the growth of the cell, cytoplasmically it will increase, and that is something that is continuous, right? Continuously that cytoplasmic growth will be there, right? Continuously that cytoplasmic growth will be there. So DNA synthesis occurs only during one specific stage in cell cycle, that is the S phase. So replicated chromosomes are then distributed to the daughter nuclei by the series of events during this division, right? So everything is under the genetic control. Everything is under the genetic control. Chalo. So before going forward, I'll tell you about this division and then we will discuss it further. Dekho, we talk about two type of divisions, right? Firstly, let's talk about the cell cycle. So in the case of cell cycle, I told you this thing clearly, right? It is a sequential, right? Sequential events are going on. Events are taking place in a sequence. Yeah, orderly sequence is there, right? So when we talk about the cell cycle, we divide cell cycle in two phases. One is interphase, another is bache M phase. One is interphase, another is M phase. One is interphase, another is M phase. Interphase is also known as resting phase, preparatory phase, energetic phase, I phase, invisible phase. There are different names for this interphase. So it is the resting phase, preparatory phase, energetic phase, invisible phase, I phase. Take care, bache, I phase. These are the different names, right? These are the different names, and sometimes it is also known as non-dividing phase या initial phase, right? Sometimes we also call it as non-dividing phase or the initial phase. These are the different names of this interphase, right? Resting stage. I'll tell you why do we call it as resting stage. And this M phase, where actual cell division occurs. So cell division will take place in this particular phase, right? So here it is the resting phase. It doesn't mean that cell is not doing anything. No, cell is preparing itself for the division. So it is the division that is the actual phase. It is the division that is the actual phase. And M here means mitosis. M here means mitosis. Okay. So firstly, let me explain you the concept here. So this phase is the actual division phase and it is a preparatory phase. So I'll explain it in detail. So basically, bache, we have two words. When we talk about this division, now we have two words. One is mitosis, another is meiosis. One is mitosis, another is meiosis. One is mitosis, another is meiosis. So when it is the mitosis, so it is the equational division. What's the word that we use for this particular division? Equational division. And when it is meiosis, we call it as reductional division. What do we call it, bache? We call it as reductional division. So one is equational division, and another one is reductional division. Right, bache? So equational, what is the meaning here? In this division, daughter cells will have same number of chromosomes like parent cell. Daughter cells will have same number of chromosomes like parent cells. Right? What is it? It is mitosis. What is it? It is mitosis. Right? And it is the equational division. It is the equational division. So daughter cells will have same number of chromosomes just like parent cell. Is that clear? Tell me, is that clear? Tell me. Okay, so daughter cells will have same number of chromosomes like parent cells. Just let's say it is a diploid cell. It is dividing into two daughter cells. So daughter cells will also have the same number of chromosomes just like parent cell. So daughter cells are also diploid. I'll give you one example. That's why it is the equational division. Now let's say parent cell is haploid. So if they will undergo mitosis, the daughter cell is also going to be haploid. Are you getting my point? So in the mitosis, basically, right, from one cell division, two daughter cells will form. How many daughter cells will form, bache? Two daughter cells will form having same number of chromosomes just like parent cell. This is we are talking about the actual division phase. So we are talking about the mitosis. This is what mitosis is. Now when you talk about the meiosis, it is the reductional division. It will reduce the chromosome number to half. Our nature, right? Nature is taking care of everything. Nature is like, let's say, if two diploid cells, they will fuse with each other. They will form a tetraploid individual, right? Imagine that tetraploid individual, its gamete, if there is no meiosis, they will also be tetraploid. Then they will mate, right? Some octaploid individual will be there, right? But it should not be like this. The chromosome, the DNA content, like more number of chromosome, yeah, DNA content, obviously it is going to change the appearance of that individual. You cannot say that organism will be the human being only. No, here we are retaining that thing. Right, bache? So we are having, if there is no defect, we all are having 46 chromosomes, right? Our nature is, you know that nature believes in balance, right? So they have maintained everything. Now when we talk about the meiosis, which is the reductional division, bache, it occurs in 2 rounds. Obviously in today's lecture also, we'll discuss it in detail. It occurs in 2 rounds, right? Because we have to reduce the chromosome number to half. So remember, meiosis will always be there in diploid cell. It will always be there in a diploid cell, right? But mitosis can occur in diploid as well as in haploid cell. Matlab here it doesn't matter, but meiosis will always be there in diploid cell. Now this diploid cell, it will undergo meiosis 1. Matlab it is taking place in 2 rounds. It will undergo meiosis 1, and that meiosis 1 will reduce the chromosome number to half. So it will form daughter cells. It is going to form daughter cells having haploid number of chromosome, half the number of chromosome than that of the parent cell. Then bache, there will be meiosis 2. As I said, there are 2 rounds of division. How many? There are 2 rounds of division. So meiosis 1 is there, meiosis 2 is there. Meiosis 1 is there, meiosis 2 is there. Meiosis 1 is there, meiosis 2 is there. Right, bache? Meiosis 2 is there. After that, what are we going to get? We'll get 4 haploid, right? 4 haploid daughter cells, right? 4 haploid daughter cells. So in mitosis, there will be 2 daughter cells having same number of chromosome like parent cell, and in mitosis, there will be 2 daughter cell having same number of chromosome like parent cell, and in meiosis, there will be 4 haploid daughter cells. There will be 4 haploid daughter cells. Is that clear? Is that clear? So that is what will happen in the division phase, in the M phase. This is the division. We have to discuss it. And then I told you about the interphase. So yes, before starting this M phase, we have to understand what interphase is and why do we call it as a resting stage, right, bache? And what is the importance of this interphase? Definitely we have to discuss that. But before going forward, I would like to discuss what about the genome, right? About the genome. Chaliye, I will take the example of humans only. Again, I will take the examples of humans only. So we are in our diploid state, we have 23 pairs of chromosome. That is in totality, we have 46 chromosome, right? But we have already covered the chapter cell. We know about the chromosome. It is also a chromosome. Don't be like it is a chromatid. No, it is also a chromosome. It is a chromosome. Here in this particular chromosome, this part is primary constriction. It is what? It is primary constriction. What's that, my dear students? It is primary constriction. So what is the another name of this primary constriction? It is centromere. What is the another name of this primary constriction? It is centromere. Are you getting my point? It is centromere, and obviously it is a chromosome ultimately made up of DNA and proteins only. This arm, this particular side, ultimately what does it contain? The DNA and the protein only. This is your chromosome. Don't be like it is not chromosome, it is chromatid. It is also chromosome. Primary constriction. ठीक है जी? And it is also a chromosome. In today's lecture, I'll tell you the difference in two. I will tell you the difference in two. So again, it is the centromere only, primary constriction. So these two arms, they are attached to the same centromere. So here we use the word sister chromatids for it. Sister chromatids for it, right? I hope you remember what chromatin is. They say DNA is coiling over the histone proteins. DNA is coiling over the histone proteins. Yeah, DNA plus protein together, they are making what? They are making chromatin, right? They are making chromatin, right, bache? They are making chromatin. Is that clear? Is that clear? Sure? Sure? Pakka? Right, so sister chromatids are there. This is the centromere. ठीक है जी? That is what we have. So both are chromosome. I'll explain the difference. What is the difference in these two? I'll explain it. Now there is one term that is known as homologous chromosome. Homologous chromosomes. Homologous chromosomes. जैसे I told you we have 23 pairs, right? 23 pairs. So बच्चे homo means same. Homo means same. Right? What is the meaning of homo? Homo means same. So anybody in the class who can explain, who can just write the definition of the homologous chromosome? It's something very simple. It's something very simple. Right? Homologous chromosome. Yes? Right, bache? homologous माने homologous chromosome. Homo means same. So it's a pair of chromosome. First of all, it's a pair of chromosome. It's a pair of chromosomes in which one chromosome is contributed by one chromosome is of mother and another one is of father. Okay, so is it the definition? No. When you are talking about the homologous chromosome, it's a pair of chromosome, right? It is a pair of chromosome in which both the chromosomes, right? It's a pair of chromosome. Again, I'm specifying it's a pair of chromosome, right? What is it? It is always in a pair. It is there in the diploid organisms, right? It is there in the diploid organism. It is in the form of it is basically a pair. What's that? Yes. What's that? It's a pair. Just a minute. One minute, huh? So it's a pair of chromosome having same length, same centromere position, the same size, same centromere position, same length, same length, or you can say that same size, same shape, same centromere position, and genes at same genes at same locus. Right, matlab they are having the same genes at same, matlab that position is also specific. Let me explain that to you. Right, even that position is also specific. Let me explain that to you. Okay, bache? Right, so that genes are with the same या corresponding loci. I'll explain that to you as well. Just a minute. Right, 1 minute, guys. Right, so they are having the genes corresponding to same locus. Yeah, locus is singular, loci is plural. Now let me explain that. Homologous chromosome. So it is the pair of chromosome. Just say, let's say this is a homologous pair of chromosome. Right, let's say this is a homologous pair of chromosome. So the point here is that they are having same shape. Right, the point here is that they are having same shape, they are having same size, right? Yeah, you can say that they are having same length, they are having same centromere position, and same genes are present. Yeah, genes, they correspond the same locus. What is the meaning of that particular line? First of all, when you look at this, as I said, it is in the form of a pair. One is from, let's say, from mother. Right, for females we use this symbol, and for males that will be the symbol. So let's say mother is giving one and father is giving another. Right, so the position of gene on a chromosome, right, that position of a gene on a chromosome, that position, for that we use the word locus. For that we use the word locus. In a very simple language I'm trying to explain it to you. Right, bache? In a very simple language I'm trying to explain it to you. It is the locus. Locus means position of gene on a chromosome. So plural is loci. Plural is loci. Now when you talk about the homologous pair of chromosome, right, now when you are talking about the homologous pair of chromosome, just say, let's say if here, just giving you one random example, let's say if here there is a gene for height, I'm just giving you any random example. Just say, no doubt in the case of humans, you know that polygenic inheritance is there when we talk about the height. But now let's not enter in the details. Let's understand the concept here. Just say, suppose here there is the gene for height. So on the another organism also, here you are going to see the gene for height only. Right, bache? Here you are going to see the gene for height only. Are you getting my point? Are you getting my point? Right? Are you getting my point? Right? Yes. Just let's say if here there is a gene for eye color, here also you will see the gene for eye color. So that doesn't mean that they are identical. You cannot say that homologous pair of chromosomes, they are identical. No, they are not identical. They are not identical. All I'm saying is that they are having same shape, same size, same centromere position, right, bache, and genes with corresponding, okay, genes with corresponding same loci. So basically that same gene position is also there. If here there is a gene for character A, here also character A, here for character B, here also character B. So gene, that position is same, but alleles can vary. Alleles can vary. Matlab, it is not like that it will totally be same. The position of gene, that locus will be same. If here there is a gene for the height color, here also you are going to see like that, but the allele, right, but different alleles can be there, right? So this allele concept we discuss in the class 12th, we are going to discuss that in the class 12th, but still, just say in class 10th also, if you are in class 11th, then you don't know about it, right? Matlab, you maybe you know some things related to it because it is there in class 10th, but if you are a dropper, you understand this thing, right? So when we say gene, we say that it is a pair of, it's a pair of allele, right? So that allele is nothing, it's like the same gene, but it is having some mutation. That's why some changes are there. Just say, let's say if this is a pair of gene. So here you will see one allele is for dominant character and one allele is for recessive character, and when they are present, because dominant is going to express itself, that plant will be tall only, right? So it is in the pair. So obviously, let's say if this is your one chromosome, this is your another chromosome, let's say one is the paternal chromosome, another is the maternal chromosome. If here you have the gene for height, here also you will have the gene for height, but allele can vary. Just say here if it is capital T, here small t can also be there. Okay, bache, here small t can also be there. Is that clear? Here small t can also be there. So allele can vary. Are you able to get it? Yes? Yes? Right? So now you must be thinking ki why are you talking about the homologous pair of chromosome? Right? Why are you explaining this to us? You were talking about the genome. So bache, because I was telling you about the genome and I told you the haploid content of a cell is basically, right, haploid content of the cell is the genome. Haploid content of the cell is genome. So what is the meaning there? Dekho, let's say if it is a homologous pair of chromosome, this chromosome is telling me ki here you have gene for character A, for character B, for C, for D, right? So obviously this, because it is in a pair, it is in a pair, so it is the homologous, right? These are the, this pair is the pair of homologous chromosome. So obviously here on this chromosome also there will be the gene A, B, C and D, right? It is going to be the same. Allele can vary, but ultimately gene is going to be the same. Yes or no? Yes or no? Yes or no? Please tell me yes or no. Done? Done? Is that clear? So if this chromosome is having genes for ABCD, this is also having genes for ABCD. So even if I'll study this one, I'll get the same information. Even this is also going to give me the same information. So when we are saying the genome, no doubt it is a genetic information of a cell. So it is a haploid content of a cell. So out of 46 chromosomes, if I will pick up that 23 chromosomes, that will give me an idea that which genes are present there, right? Obviously in the case of boys, because X and Y, these two chromosomes are different. So in that case, we have to study X and Y both, right, to know about the genes. But otherwise it is like that. Are you getting it? Are you getting it? Are you getting it? Yes? That why am I relating it with the homologous pair of homologous chromosomes? Right? Right? So it is important, keep it in your mind. Okay, chali, done. So this part clear. So now we have to start the phases of cell cycle, which is interphase and M phase. And now we have to discuss that why interphase. Why interphase is the resting phase B, it is the energetic phase B, it is the invisible phase B. Why is it so? Let's discuss that. So are you ready? Yes, are you ready? Tell me, students, are you ready for that? Okay, देखो, interphase and M phase. This is my famous example, and I don't know whether it is my famous example or not, but it is my favorite example. Right, interphase or M phase, these are the two words that we have. Right, interphase and M phase, these are the two words that we have. देखो, it is just like your NEET preparation. It is just like your NEET preparation. How, ma'am? देखो, if you want to crack NEET examination, you have to be, after 10th you'll move to class 11th, then class 12th, right? I'm not talking about that thing, dummy school or this. Obviously, at least you have to give 2 years, right? You have to give 2 years. Then you will appear in that NEET examination. So some students after 2 years they appear, or after 2 years they are able to crack. Some students, they even take the drop years, right? They are the droppers. Some even they take 1 year, 1 drop year, 2 years, 3 years. It can be anything. Why? Because they are preparing. They are preparing for what? They are preparing for this particular exam. Now this exam is for 3 hours 20 minutes, right? This exam is for 3 hours 20 minutes. And if you want to crack this exam, you have to complete your syllabus. You have to give at least 2 years. You have to give at least 2 years, right? Then obviously if it goes well, then you'll get admission. Otherwise you have to prepare again. You know it very well, isn't it? You know it very well. So this phase is your preparatory phase. You are preparing. Why? Because you want to give your best here in 3 hours 20 minutes. So if you want to give your best, the best in this 3 hours 20 minutes, you have to give your best in your preparatory phase. It is not possible that you are not preparing sincerely and you will be able to crack the examination. Of course not. Of course not. So you have to prepare sincerely, right? Then only you will be able to crack this exam. So just imagine you are giving your 2 years, 3 years for this 3 hour 20 minute ka exam. Right, same is the case here. So in your preparatory phase, right, for other people, for your relatives, even sometimes for your family, they are like, "Arey, they are doing nothing, they're just preparing." Right? We hear things like that. Sometimes even our parents, our siblings say, they are doing nothing, they're just preparing. They're just preparing. But we know that we are under immense pressure when we are preparing. Right. We are under immense pressure when we are preparing. We have to give our best. We have to make sure that we don't waste our time. Why? Because if we'll waste our time, we are going to suffer here on this D day. We are going to suffer. Right? We are going to suffer. So for other people, you are doing nothing, but you know that you are under immense pressure. You have to complete the syllabus. You have to make the notes. You have to, do that, solve that MCQs. You have to give the mock test, right, bache? But other people they cannot realize it. Others they cannot realize it. For them, you are just preparing, right? You are just sitting in front of a laptop, right? You are just attending a YouTube lecture, or you're just going to some offline class, and then you are coming back and you're reading. But you know that it's very important, right? Why? Because you have to give your best. So same is the case here. When we talk about the interphase, it is the preparatory phase. It is the invisible phase. Because here also your hard work is invisible. Only your result will make it visible. You know that now? So in this interphase, a cell is preparing itself for the M phase, right? A cell is gathering everything possible required for the cell division. Because cell division is energetically a very expensive process, right? It is energetically a very expensive process. So actually when we see the interphase nucleus under the microscope, so it will just appear like this. This is a cell and some threads are there, right? Obviously when a cell is resting, right, the chromosomes will not attain, the chromosomes are not in this shape. When a cell is resting, chromosomes are not in this shape. They are not compact, right? That there will be the chromatin only, that threads, The DNA and coiled over the proteins, that's all. That thread-like structure. So when we see the interphase nucleus, interphase cell in interphase stage under the microscope, we are going to notice nothing, right? We are not going to see anything. We'll be like, yeah, some threads are there. That's why sometimes it is referred to as resting stage, right? But it is the energetic phase. It is the energetic phase. It is the preparatory phase. It is the phase where cell is metabolically very active. Metabolically it is gathering, it is performing that metabolism so that it can have everything possible required for this M phase. So all the protein synthesis, all the essential proteins, RNAs, that nucleotides, the enzymes, right, the tubulins, whatever is required, even the DNA replication, all of it will occur in the interphase. Why? Because they have to make sure that there is no error in the M phase. We want this M phase, this division phase to be error free. Right, whatever is required for this division phase, it should be available there in the cell, right? So that is how you can relate. And obviously, if you compare your preparation time with the exam duration, so you are spending most of the time in this preparatory phase, same is the case here. So when you talk about a cell is going to spend more than 95% of its lifespan, more than 95% of its lifespan in I phase. Are you getting my point? In interphase. Are you getting my point? It is going to spend more than 95% of its lifespan in interphase. Are you getting my point? Yes. So actual division phase is M phase. Okay. Done? Sure? Right? So let's say in the case of humans, right? So within 24 hours, you will see in 24 hours that human cell, for 23 hours, that human cell is in the interphase, and for the division phase is 1 hour. So this duration of cell cycle and all, it can vary. It can vary from organism to organism. Just in the case of yeast, it is for 90 minutes only. Right, bache? In the case of your E. coli, that is your Escherichia coli, it is 20 minutes. Right, so one cell is dividing into two, right? So this time period where one cell is dividing into two, it is the generation time. It is the generation time. So is that clear? Is that clear? So let's discuss it, bache. So here you guys can see, okay, let's highlight it from the NCERT, that a typical eukaryotic cell is illustrated by human cells in culture, right? It is illustrated by human cells in culture. So see, these cells, they divide once in approximately every 24 hours. These cells, they divide just once in approximately every 24 hours, right? So the duration of cell cycle can vary from organism to organism. So in yeast, it is for 90 minutes only. So what is yeast? Unicellular fungi. It belongs to the class Ascomycetes. Right, bache? It belongs to the class Ascomycetes. Right, bache? So interphase and M phase is there. So M phase represents the phase when the actual cell division or mitosis occurs, and the interphase represents the phase between two successive M phase. A very wonderful line, and that's how I'm going to discuss it. So interphase is a phase between two successive, one after another, M phase. Okay, two successive M phase, we'll discuss it. Now, bache, in the interphase, so here देखो, human cells, in the 24-hour average duration of cell cycle of a human cell, the division lasts for only about an hour, and interphase is for more than 95% of the duration. I hope it is clear. Okay? Now, bache, when you talk about the interphase, remember this thing. In the interphase, in the resting state, because the DNA replication will also occur, the cytoplasmic increase is there. So here you are going to talk about G1S and G2 phase. What are we going to discuss here, bache? What are we going to discuss here? We will talk about the G1 phase here, we are going to talk about the S phase here, and we are going to talk about the G2 phase here. Okay, G1 phase, S phase, and G2 phase. Right, so G1 means growth 1, your first growth phase, sometimes gap 1 phase it is. Right, now one more thing, and this G1 is also known as post-mitotic phase. You people should know the another names, right? It is also known as post-mitotic phase. It is also known as post-mitotic phase. So G1 phase, growth 1 phase, gap 1 phase, post-mitotic phase. Now this S phase, bache, S phase, synthesis phase. Synthesis phase. It is the phase where DNA duplication or DNA replication occurs. So DNA replication is specifically, it will take place in this phase only. Right? DNA replication, even centriole duplication, we'll talk about it. Then G2 phase, growth 2 phase, gap 2 phase, pre-mitotic phase. Sometimes we can also call it as post DNA synthesis phase. Matlab it is taking place after DNA synthesis. So post DNA synthesis phase it is. Okay? And this one, this G1 phase is pre-DNA synthesis phase. Okay, ji? So this is the interphase G1, S and G2. Right? And when you talk about the M phase, the actual division phase, don't worry, I'll write it properly as well. So here you have two things, karyokinesis, cytokinesis. Karyon, nucleus, kinesis, division. Here you are talking about nuclear division. Here we are talking about nuclear division, and that is followed by cytoplasmic division. That is followed by cytoplasmic division. Is it clear? That is followed by cytoplasmic division. Is it clear? Is it clear? So we have interphase, we have M phase. These are the phases of cell cycle. Please note down. Done? Yes? Yes? Bache, which doubt do you have? Tell me. Tell me. Please mention it. Right? Do you have any doubt? Please tell me. Chaliye. So now I just use one line. I just mentioned one line that is obviously from the NCERT. That, and this line is saying ki interphase is a phase between two successive M phase. Right, bache, please type your doubt. Interphase is a phase in between two successive M phase. Right? Yes. Now, that's how we can write it. M phase, M phase, I phase, because we said interphase is a phase between two successive M phase. Now, this I phase is having G1 phase, it is having S phase, it is having G2 phase. And this M phase is having what? It is having karyokinesis. Firstly, there will be the nuclear division and there will be the cytokinesis, cytoplasmic division will be there. Here also, firstly, there will be karyokinesis and then there will be cytokinesis. Okay, bache? Now this karyokinesis means nuclear division. Here we have a trick. It is PMAT. What's the trick that we have, bache? PMAT. What's the trick that we have here? Yes. What is the trick that we have here? It is PMAT. Please mention it in the chat section. Yes. Please mention it in the chat section. It is PMAT. Matlab prophase, metaphase, anaphase, telophase. So during nuclear division, that's what we are going to discuss. Prophase, metaphase, anaphase, telophase. So in this sequence only you have to remember. Now now please observe this thing. See this G1 phase. My dear students, G1 phase. See, it is as per NCERT. Ki interphase is the phase between two successive M phase. So it is the G1 phase. Don't you think that G1 phase is coming? Matlab from this line, what can you conclude? That G1 phase is coming after this M phase. G1 phase is coming after this M phase. So can I call it as post-mitotic phase? Yes or no? You tell me. Can I call it as post-mitotic phase? Yes or no? After mitosis, we have the G1. And now, here, it is the G2 phase. It is before this M phase. So, it is pre-mitotic phase. It is pre-mitotic phase. It is pre-mitotic phase. Now, when we talk about the S phase, right? It is pre-DNA synthesis phase. Post DNA synthesis phase. Done? Right? So it is your, now I hope you are able to understand this. It is your S phase in which actual DNA replication occurs. Right? Right? The initial amount of DNA will get doubled and even the centriole duplication. It is the favorite question of your examiner, right? Even the centriole duplication will take place here, right? Even the centriole duplication will take place here. Now is it clear? Now is it clear? Please note down. Yes. Done? Chaliye, note down, then we'll start the next part. Okay, chaliye, let's go. Let's discuss this. Now, there is one more phase that we have to discuss, that will be G0 phase. We will cover it, but up to this part all clear? Yes, tell me, up to this part all clear? All clear, bache? So let's complete it from the NCERT also. So you can see here, the M phase starts with the nuclear division corresponding to the separation of daughter chromosomes. ठीक है जी? And usually end with the division of cytoplasm. So interphase, though called the resting phase, it's a time during which cell is preparing itself for the division by undergoing both cell growth as well as DNA replication. So DNA replication will be there in the S phase, and that too in an orderly manner. So G1 phase, S phase and G2 phase, right? These are the three phases in the interphase. Chaliye, now let's talk about these phases. Okay, and after completing the cell cycle part, right, after the completion of the cell cycle part, bache, I will give you a break. Okay, I'll give you a break. Done? And in this particular chapter, we have to talk about the cell checkpoints. It is not given in NCERT, but yes, question can come from this part. So G1 phase, S phase, G2 phase. Yes, G1 phase, S phase, G2 phase. Right, G1 phase, S phase, G2 phase. Write down and make the table like this. Then we will write all the important points here. Quickly do it. Just a minute. Quickly do it. Quickly. Done? Chaliye, let's discuss this. Okay? Okay. So the word is G1 phase. Ultimately, bache, in these phases, the cytoplasmic increase will be there. And always remember, whatever is required for the S phase, that will be prepared in the G1 phase. ठीक है जी? मतलब like whatever is required in the S phase, that will be formed in the G1 phase. So S phase is mainly for what? For DNA replication. ठीक है? So when it comes to the G1 phase, can I say that it is the phase? This is the G1 phase. Can I say that it is the phase between end of mitosis and beginning of DNA replication? Just look at it. It is the phase in between the end of mitosis and the beginning of DNA replication. Can I say so? Can I say so? You tell me. Can I say so? Obviously, ma'am. So it is the phase in between. It is the phase in between the end of mitosis and beginning of DNA replication. Yes, beginning of DNA replication. Right? Yes, bache. So next is synthesis of 3 types of RNAs. 3 types of RNA matlab all your rRNA, mRNA, right, tRNA. So synthesis of 3 types of RNAs, enzymes, proteins, nucleotides, everything will be done here in this particular phase. Yes, bache. Right. Even the content, amino acid, cell organelles, matlab their content will also get duplicated. Okay, done, bache. Is it clear? Yes. So cell is here. Cell is what? Cell is metabolically active. Cell is metabolically active here. and continues to grow. Cytoplasmic increase is definitely there. It continues to grow. Is that clear? It continues to grow. Right, bache? Right. So most of the DNA organelle duplication, most of the organelle duplication occurs here. Most of the organelle duplication occurs here, right? Done. So chromosome has single chromatin occurs, single chromatin thread. So basically it is monad in nature. Chromosome has single chromatin thread. I'll explain this. So basically monad in nature, right? This is going to happen in the G1 phase. This is going to happen in the G1 phase, right? So it is ultimately doing everything, it is growing, but DNA replication will not occur. DNA replication will not occur, right? Now, one more thing. When you talk about this G1 phase, now, it is the most, its time duration is variable. Variable time duration. Matlab, you cannot say that this phase will last for 20 minutes, 30 minutes, 1 hour or 2 hours. Its duration is variable. It depends upon the need. Let's say if there is a cell, it has to divide very quickly. The G1 phase, its duration will be reduced. So this G1 phase, when you are talking about the G1 phase, its duration is not fixed. Right, bache? Its duration is not fixed. So it is most variable in length. It is most variable in length. Is that clear? It is most variable in length. This point clear? Yes. This point clear? It is most variable in length. ठीक है जी? Variable time duration, G1 phase. Now in the G1 phase, there is something very beautiful, right? We know that after G1 there is S, there is G2. Are you aware of it? There is after G1 there is S, there is G2. Are you aware of it? Actually, bache, in the late G1 phase, let's say somewhere here, there is something known as kind of restriction point. Okay, there is something known as restriction point. Animal cell mein it is restriction point. In the yeast cell, it is a kind of start point, right? So when a cell is in the G1 phase, if that cell has crossed this restriction point, matlab it will enter in the S phase, right? It will enter in the S phase. Yes, bache? Yes, bache. So in the cell cycle, there is a regulatory or restriction point. There is a regulatory or the restriction point. Right, bache? Right. If there is a go ahead signal, if there is a requirement for that cell to divide, if it is required that this cell has to divide, if our body is giving that go ahead signal, okay, obviously various factors are there which are going to do that. So there is something called restriction point which is present here in animal cells. The cells in G1, right, in the late G1 phase, there is a restriction point. If a cell will cross it, right, it will proliferate. It will move to the S phase. It will move to the S phase. Are you getting my point? It will move to the S phase. Right, bache? Right. So progression, proliferation is not the right word. Progression from G1 to S. So if a cell has crossed this restriction point, there is the progression of the cell from G1 to S. It is a PYQ also that after entering in which phase the cell will continue the cell cycle or something. There is a question like this. Okay? So this restriction point, it was first discovered in East, bache, and there it was known as start point. Right? In East, we don't call it as restriction or regulatory point, we call it as start point. So animal cell mein you can call it as restriction point. So once it will cross it, so there will be the progression of this cell from G1 to S. But let's say, if there is no requirement for that cell to divide, if there is no requirement for that cell to divide, it will enter in a G0 phase. Jaise ye dekho. So if cell has crossed this restriction point, definitely it will go to the S phase and it will continue the cell cycle. But if there is no go ahead signal, it will enter in the G0 phase. Jaise ye dekho here. They made this arrow na G naught phase. So this G naught phase is also known as bache cell arrest stage. It is also known as quiescent stage. We can call it as quiescent stage or the cell arrest stage. Now, why is it so? Because the division of that cell is not required. Just I'll give you the example of our neurons. We say na neurons only divide at the time of birth, only the neurons will form. Later on, they do not divide further. Yeah, heart cells. Okay, bache. So actually, there is a study in which it was said that even neurons also divide, but they take a lot of time, yeah, many years to divide. But right now, I'm not able to it was just a kind of research was going on, right? So we do not have that proper data regarding that. So that's why I'm not discussing it that in detail, but that is also the point. Okay? So we are talking about what? We are talking about the neuron cells. They say they will divide only at the time of birth, yeah, when that zygote is growing. So obviously, so because they will not divide further, so where are they present? In the G naught phase, right? And they are permanently in the G naught phase. Matlab this G naught phase for a cell can be permanent, it can be temporary, right? So this neurons, ya fir heart cells, because they are not dividing, they are in G naught phase, they are in this quiescent stage, right? But let's say if now you don't need a cell to divide, it will enter in G naught phase. But later on you want this cell to divide, then obviously there will be some growth factors which will be provided to this cell. This cell will again enter in G1, again it will divide. Right, so in that cell's case, it will be temporary hold, yeah, temporary, it is temporarily there in the quiescent stage. Is that clear? Is that clear? Yes. Is that clear? So when it is the G naught phase, cell will remain metabolically active. There is no problem with the metabolism. Cell remains metabolically active. Metabolism is going on. Metabolism is important as well. Metabolism is important as well. Cell has to show the metabolism. If metabolism is there in a cell, it means cell is living. Cell is living. Metabolism and being living is something like in biomolecule it is written that these two words are synonyms. Right? If there is a living organism, definitely chemical reactions are going on. If there is a living organism, definitely chemical reactions are going on. Okay, bache? So cell remains metabolically active. Metabolism is there, right? It's not like some inactive state, some dormant stage where cell is doing nothing. Metabolism is there, right? Whatever chemical reactions are required, it is there. So cell remain metabolically active. It's just that, but do not proliferate. Proliferate means it will not divide into two. Take care. So, but do not proliferate unless they are called on to do so. Is that clear? Okay. Sure. Right. So it depends upon the requirement of the organism. Done, bache? So some cells they do not divide. Some cells they divide occasionally. Okay. Some at the time of injury, when this is some cells they are not dividing, but at the time of injury we need that cell growth. So obviously they will leave the G naught phase. They will again enter in the G1 and they'll continue the cell cycle. So it is like that. Okay. It is like that, okay? So G1 phase is also clear. This duration thing is also clear. This monad and dyad thing I will explain, but tell me if it is clear to you or not. Yes? ठीक है. The main phase, S phase, DNA replication occurs. Right, and monad changes to dyad. ठीक है? Monad changes to dyad. I'll explain you the meaning. Let's say if initial amount of DNA is 2C, I will explain it in detail. Please write down. Then final will become 4C after this. Final will become 4C after it. ठीक है? S phase. Plus centriole duplication is also there in the cytoplasm. So it is the favorite question of the examiner. They ask it most of the time. So centriole duplication will also occur in S phase. Are you getting my point? Are you getting my point? Right. So synthesis, because histone proteins are required for the packaging of DNA. So remember, synthesis of histone and non-histone proteins occur here. Okay, it will occur here. Now, I'm going to tell you the most important point. Are you ready for that most important point here? Tell me. I am going to tell you the most important point here. Are you ready for that? Yes, please. Are you ready for that? Are you ready for that? Sure? Sure? So the most important point is that DNA amount will get doubled, but the number of chromosome will remain same. Okay? That it is the DNA amount. DNA amount will get doubled, but number of chromosomes remain same. It is the DNA amount that will get doubled, but number of chromosome will remain same. It is a very important point. I will discuss with you all that why is it so. Firstly, write down. Okay? Okay. Okay. Right. So in the S phase itself, let me make it here. बच्चे kinetochore subunits will also assemble. हाँ जी, in the S phase itself now, kinetochore subunits they assemble and chromatids attached to centromere. Sister chromatids attached to centromere. You write it down, please. Okay, so kinetochore subunits they assemble and now sister chromatids attached to centromere, right? Right. And I already told you about the duplication of the centriole. Okay. Now what is going to happen in the G0 phase? I'll come to this point. Why DNA amount is getting doubled but the number of chromosomes is same. So in the G0 phase, what's the story, bache? Again, cell growth continues. It continue. Cell growth continue. Right. Tubulin. Do you know tubulin? Yes. In the last class also we discussed that. Tubulin. Do you know tubulin? Yes. Do you know what tubulin is, students? Yes. Tell me, guys. Tell me, please. Do you know what tubulin is? I'm expecting an answer, guys. Do let me know in the chat section. Be quick. Do let me know in the chat section. Very good. So tubulin is a protein. It is required. It's polymerization. Alpha, beta tubulins, they will polymerize. They will form protofilament of microtubules. Many protofilaments together will form microtubule. Matlab, in simple words, tubulin is required for the formation of microtubules. Tubulin is required for the formation of microtubule. So tubulin synthesis occurs here. Right, tubulin synthesis occurs here. ठीक है बच्चे? Okay. So cell growth continues. Right, the cell energy stores will increase. increases in this phase, that protein synthesis and all, it is going on. ठीक है जी, it is going on as such. So cell का size, energy store, even the nucleus increases. ठीक है जी. It increases. ठीक है. But now what is the point that we need to understand? That why do we say that in S phase DNA amount get doubled, but number of chromosome remains same? It is the DNA amount that will get doubled, but the number of chromosome will remain same. Why do we say so? Why do we say so? I'll tell you. देखो बच्चे, when we draw this type of chromosome ना, मतलब एक, this is a compact structure. This is a very compact structure. It is only visible, that type of chromosomes are visible after prophase. मतलब in the prophase the condensation will start and we will see that type of chromosome. But because now I have to explain you something, right? Now I will use this compact form only. ठीक है? I will use this compact form only. जैसे I told you initially that this is also a chromosome, this is also a chromosome. It's just that it is monad. It is having single arm. It is having two arms, right? It is having these two arms. We are calling it as sister chromatid, right? We are calling it as sister chromatid. So that's why we are using the word dyad here. That's why we are using the word dyad here. Single arm is there. It is the monad. Actually, what's the story? I'll give you the example. Ultimately, you know that what chromatin is. Your DNA with proteins, it is forming the chromatin. Your DNA with protein, right? Your DNA with protein, it is forming the chromatin. Yes, bache? Yes, bache? Yes or no? Yes or no? Hi, Mahran ji, I don't know about your question. Please ask it again, then I will definitely try to answer your doubt. And trust me, if I don't know that, I'm going to tell you that thing clearly. Okay? Don't worry about that, Mr. Mehran. Okay? Chalo. So this is monad. This is sister the dyad. Usually you know that, and especially in the interphase, your DNA will be in this form, right? Matlab it will be tangled, it will be in the form of thread. It is not going to show the compact structure, but because I have to explain you this chromosome number thing, so let's assume, let's say this is a cell. This cell is having how many chromosomes? They are having 4 chromosomes. In its diploid state, let's assume that this cell is having how many chromosomes? 1, 2, 3, 4. 4 chromosomes are there. How many chromosomes are there? 4 chromosomes are there. Right, bache? How many chromosomes are there? 4 chromosomes are there. है ना? Let's assume. Again, I'm telling you, in the G1 phase, your chromosomes are not going to be like this, but you just need to believe, you just need to assume it here. ठीक है? Though they are in the thread-like form and they are having single arms. ठीक है जी? 1, 2, 3, 4. After S phase, as I said, the DNA replication occurs, the DNA duplication occurs, and what do we say there? That 2C, which is the initial amount, it will become 4C. What is that 4C? Final amount of DNA. But how is it possible? Right, देखो, simple. जैसे this chromosome is ultimately made up of what? It is having the DNA and proteins only, na? DNA. So in the S phase, the DNA will get doubled. So that is how the things are going to be, right? That matlab 2 sister chromatids will form. 2 sister chromatids will form actually in the S phase. Means if it is, let's say if you are considering it is in the thread form, let's say if it is something like this. So after S phase, it will be this. This, right? So basically number of chromosome is still same, 1, 2, 3, 4. But see, it was monad here. Single arm was there. Now it is dyad here. 2 arms, matlab 2 sister chromatids. They are attached to, why do we call them as sister chromatids? Because they are attached to the same centromere, right? So number of chromosomes is the same, but the DNA content is double. Let's assume initially if it was 100, After S phase, it will become 200. Yeah, in S phase, it will become 200. Are you getting my point? Are you getting my point? Yes or no? Are you getting my point? Right. So if 100 picogram, then it will become 200. So number of chromosome is still same, but the DNA content is getting doubled. So in S phase, actually, you will see the chromosome like this, having two sister chromatids. Having what? Having two sister chromatids. Is that clear? Having what? Having two sister chromatids. Is that clear? Is that clear? Are you sure? Are you sure? Right? So number of chromosome is same, but the DNA content will get doubled. Okay, the duplication here occurs. That's why it is the diid word here. I hope it is clear. So now you know what cell cycle is, and basically in the interphase, cell is preparing itself so that it can just do well during the M phase. So now tell me, let's say if 2n, if diploid number of a cell is 16, tell me the number of chromosomes. Tell me the number of chromosome in G1, S and G2 phase. Quickly, tell me the number of chromosome in G1, S and G2 phase. I'm waiting. I am waiting for it. Tell me quickly. I am waiting for it. Tell me quickly. Yes, students, please tell me. I'm waiting. हाँ जी. Number of chromosome will remain same. So 16 here, 16 here, 16 here. So in the G1, the DNA amount will be just say initial amount 2C. Here it will be 4C. Here it will be 4C. So that's what you need to know. That's what you need to know. Okay. So see this. You have studied mitosis in onion root tip cells. It has 16 chromosomes. So you should know that onion, in its diploid state, it is having 16 chromosomes. Even you need to focus there. So how many chromosomes will be there in G1 phase, after S phase and after M phase? Right? At G1 phase, after S phase, M phase, obviously number of chromosome will be same, and you are talking about mitosis. So DNA content, right, it will be like this. Okay? Right? Now, how do plants and animals continue to grow throughout their life? How do plants and animals continue to grow all their lives? You know that in the case of plants, they can grow throughout their life because of meristematic tissue, right? But still, in the case of animals, the growth is restricted to a certain age. It is restricted to a certain age. But in the case of plants, we have the meristematic tissue, the group of actively dividing cells. Okay, so in the case of plant animals, no doubt we talk about the stem cells, which are the totipotent cells, right? These days they are being preserved, that if you know that baby suffers with any issue or something, that stem cells can be used. But overall, in the case of plant, the growth is throughout, so meristematic, right? Meristematic tissue is there. So do all cells in a plant divide all the time? It is the meristematic tissue, na? That cells will divide. They will add new cells. Do you think all cells continue to divide in plants and animals? No. In animals, you know that it is even restricted. And even like if I have to be more specific, you know that there are some cells, just in our case, you're talking about heart cell, you're talking about neurons. They are not going to divide, right? So can you tell the name and the location? So this is what we have discussed, and you are going to answer that to me. Okay? And this part of, let's revise it quickly from the NCERT, and then let's start the mitosis. So G1 phase corresponds to the interval between mitosis and initiation of DNA replication. Is it clear? Is it clear? Is it clear? Hanji, is it clear? So during G1 phase, cell is metabolically active, continuously grow, but doesn't replicate its DNA. So DNA replication is there in the S phase, right? So initial amount of DNA, if we are denoting it as 2C, then it will increase to 4C. DNA amount will get doubled, but no increase in the chromosome number. No increase in the chromosome number, a very important point. So if a cell had diploid or 2n number of chromosome at G1, even after S phase, number of chromosome remains the same, that is 2n. That is 2n, right? So in animal cells during S phase, DNA replication begins in the nucleus and the centriole also duplicates. So centriole, right? Even in the last class I told you, students, I told you this thing in the last class, right? That centriole is present in the animal cells. It is there in the animal cell. It is absent in the plant cell. Not in all the plants, but yeah, still. Okay, so during the G2 phase, proteins are synthesized in preparation for mitosis, right? While cell growth continues. Okay, so here the same thing is given, bache, that some cells in the adult animals, they do not exhibit division, जैसे heart cell, right? They will enter in the quiescent stage. So it is an inactive stage where cell is not dividing, inactive in context of the division, in context of the proliferation. But my dear students, overall metabolism is going on, right? So G1 phase, so this stage is overall inactive, the quiescent stage, the cell arrest stage. But that doesn't mean that cell is inactive. Metabolically it is active, metabolism is going on, but proliferation is not there. Okay, bache. Now they are saying in animals, mitotic cell division is only seen in diploid somatic cell, but in some exceptions are also there where haploid cells will also show the mitosis, जैसे male honeybee. चलो, I'll give you the example here. देखो, normally, जैसे ये देखो, 2n cells. Normally our cells are 2n. Mitosis will occur, है ना? But they have given the example of male honeybee. Male honeybee means you are talking about drone, Mr. Drone. ठीक है बच्चे, it develops parthenogenetically. Do you know that? It develops parthenogenetically. How do it develops parthenogenetically? मतलब parthenogenesis is there. What is there? Parthenogenesis is there. Yes, students. What is there? Parthenogenesis. Parthenogenesis means the development of this haploid gamete into an adult without fertilization. Without fertilization, this haploid gamete, without fertilization, this haploid gamete, obviously it is single cell, it is developing into an adult without fertilization. So obviously that cell has to multiply. No, this gamete is a single cell, it has to multiply. So that's why we are saying in animals, here you are going to see the mitosis. Here you are going to see the mitosis, right? And now when you talk about the plants, when you people talk about the plants, right? In the case of plants, what's the thing? Just say, you know, na, haplontic life cycle, right? Next lecture is of plant kingdom. Haplontic life cycle. Just say in the case of algae we see, right? So the main thallus, main plant body, main thallus, main plant body is haploid there. So if that main thallus is haploid, we call it as gametophyte, it formed gamete. So here in the case of plants, where the main plant body is haploid, so if they need to make the gametes, they'll go for the mitosis. So mitosis will occur in the haploid cells, even it is forming the gametes there. Right, bache? Right, bache? So that is the story. Right? So this is about the mitosis, which is an equational division. So next thing that we need to start is M phase. So do you need a break or can we continue? Chalo, I will give you the break, otherwise you will start crying. So, bache, so let's meet at 9 p.m. Alright? ठीक है? चलो, enjoy. Okay, so let's continue mitosis, guys. Right, so do you have any doubt from the cell cycle part? Do you have any doubt from the cell cycle part? Yes? Chaliye. So already I told you, bache, that in the mitosis, it is the equational division. I told you already. Plus, you will get two daughter cells, done. Plus, karyokinesis will be there, cytokinesis will be there. And when we talk about the karyokinesis, so it's P-MAT. What's that? It is P-MAT. Prophase, metaphase, anaphase, telophase. Right. So what's that? It is P-MAT. Prophase, metaphase, anaphase, telophase. Okay. So let's start that. Okay. Let's start that. So you see, these are the diagrams. Okay. And one by one, we will discuss about it, and we will discuss the things from the NCERT also, right? But before going forward, right, before reading the things from the NCERT, I will explain you the meaning of each and everything here. Dekho, mitosis. Before mitosis, let me tell you, there is something called amitosis. There is something called amitosis. What is there? There is something called amitosis. Matlab, it's a kind of very primitive type of division in which your chromosomes, your nucleus will divide, but there is no spindle formation and all. Right, bache? Right, there is something called amitosis. Do you know about amitosis? Do you know about amitosis? Right, it is simple division. Right, simply your nucleus is dividing, your cytoplasm is dividing. There is no spindle formation here. Right, there is no spindle formation here. So amitosis, simple nuclear and cytoplasmic division, but right, there is simple nuclear and cytoplasmic division, but there is no spindle formation. Right, proper spindle apparatus formation and all will not be there. Is that clear? It will not be there, right? And even the chromosomes will also not appear. Chromosomes mean that compact structure will also not appear. So it's a kind of direct cell division. Jaise binary fission is there, no? Jaise binary fission is there, no? Just like that. Okay? Okay. Yes. So spindle formation will not be there, and there is no condensation of the chromatin into the chromosomes, right? There is no condensation of the chromatin into chromosome. Is that clear? Now when we talk about the mitosis, see, in the case of animal cells and in the case of plant cells, right, there are differences in this mitosis as well. Although it is an equational division, right, the parent cell will divide into daughter cells. Daughter cells will have same number of chromosomes just like parent cell, but still some differences are there. Just say you are talking about what? You are talking about the animal cell. So in the case of animals, this mitosis is known as amphiestral. For that, we use the word amphiestral. For the plant cell, it is anestral. It is anestral. So in one case it is amphiestral, in another case it is anestral. Why? It is due to the centriole. Right? It is due to the centriole. Okay, so because centriole is present in your animal cell. But now, ma'am, centriole and ester, what is that? Let me explain you these general things, then we will move to the next topic. And I hope that some students are aware of it. I hope that you know it, but still I am explaining this part to you. Just say, देखो, firstly, you know that in our body two types of cells are there. We talk about the somatic cells and we talk about the germ cells. Right? We talk about the somatic cells and we talk about the germ cells. I'm explaining you some general things, then I will move to that actual topic. Somatic cell, just say your normal body cells. But these are the cells that are not forming gamete. Right? They are not forming gamete. Right? Just say, let's say You are taking your cells from your hand, from your arms, from your legs. They are the normal somatic cells. They are not going to form the gamete. Now, when I am saying germ cells, they are the gamete forming cells. What kind of cells are they? They are the gamete forming cells. They will form gametes. What are that gametes? They are the haploid cells, right? Much needed for the sexual, much needed for during sexual reproduction. They'll fuse, they'll fertilize, they'll form the zygote. That's what we know. So normal body cells they are, right? Somatic cells, they are not forming any gamete. These are the germ cells. Now somatic cells will only undergo mitosis, bache, right? With the help of that they will proliferate, means they will increase their number. They will proliferate, means they will increase their number like this. Like this, they'll form their progeny, right? Now when you talk about the germ cells, when these germ cells, when they need to increase their number, when they need to proliferate, definitely mitosis will occur. But let's say, just say we will discuss that in spermatogenesis, we will discuss that in oogenesis. But when they need to form the gamete, they will undergo meiosis. So after undergoing meiosis, after meiosis 1 and meiosis 2, ultimately we are going to get 4 haploid germ cells. 4 haploid gametes. And yes, I forgot to discuss the checkpoints. Let me finish the mitosis part, then we will discuss the checkpoints. Okay? Right? So that is the story, somatic cell and germ cell. So some students even they don't know this, so that's why I'm explaining. Now the next thing is this centriole. Even in the last class, last marathon of cell chapter, we discussed that. But a centriole is a non-membrane bound organelle. What's that? It is a non-membrane bound organelle. What is it, my dear students? It is a non-membrane bound organelle. It is not covered with, yes Arpana, it is not covered with any membrane, right? Right, and you know that it is made up of microtubules having 9+0 arrangement. I hope we are clear here. Now these centrioles, they can also duplicate in S phase. In cytoplasm, they can duplicate, right? They will duplicate in S phase. We know that. Now 2 centrioles, let's say this is the mother centriole, this is the daughter centriole. 2 centrioles, when they are covered with pericentriolar material. Remember, when they are covered with pericentriolar material, which is the amorphous substance, right? Just let's say, right, this structure. Look at this structure. Just these 2 centrioles, they are perpendicular to each other. Plus, it is covered with pericentriolar material. It is covered with what? Pericentriolar material. That is also known as meiosis. They are also 9 in number, right? So when it is covered with pericentriolar material, then it is forming a structure known as centrosome or diplosome or cell center or cell sphere. Clear? Clear? Yes. Is it clear or not? Is it clear or not? Yes or no? Is it clear or not? So centrosome, cell center, right? Cell sphere या centrosphere, right बच्चे? Is that clear? Tell me. Yes? Done, बच्चे? Yes, बच्चे? Any doubt here? This is the centrosome, cell center, या centrosphere it is, right? This is the centrosome. Okay. Now, बच्चे, when you talk about the centrosome, जैसे let's say this is the cell. Here you have the nucleus. These centrosomes na, they are, matlab, this structure is associated here, matlab, near to the nucleus, like this, right, like this, right? It is matlab basically it is, how is it present? How is it present? You can even write down that this is firmly attached to the nuclear envelope. When I'm using the word centrosome, you can simply keep it in your mind that it is firmly attached to nuclear envelope. Okay, so this part clear? This part clear? Firstly tell me. This part clear? Now, this is about the centrosome, because sometimes students, they don't know the difference in centriole and centrosome. That's why I'm telling you this. Right, bache? Now the point is, bache, the point is we are talking about the karyokinesis, nuclear division. Nuclear division. Now we will start the nuclear division, karyokinesis. But before that, this part clear? This part clear? One thing that I'm going to explain in Prophase, plus again I'm telling you, ye dekho, if this is your one centrosome, this is your another, right? From this centriole, some fibers will radiate. Some small fibers of microtubules, they will radiate. They will radiate. They will give it a star-like appearance, right? They are going to give it a star-like appearance. They are going to give it a star-like appearance. We use the word aster here. Matlab, here, when you talk about the centrosomes, right, bache? So these centrosomes, right, these centrosomes, they will radiate some fibers. Obviously, they are also the result of the polymerization of the tubulin. These fibers will give it a star-like appearance. Just say, let's say, something like this, small short fibers, right? They are not very long fibers. They are some short fibers. They are radiating from this part and they are giving it a star-like appearance, and we are using the word aster here. We are using the word aster here. Okay? And these fibers are the astral fibers. These fibers are the astral fibers. Please try to understand. These fibers are the astral fibers. So as I said, the centrioles are firmly attached to the nuclear envelope, centrosome, sorry. So they will, right, they will form these ester fibers, right? And that ester fibers are basically going to, you can say that a kind of, they are going to move these centrosomes to the opposite pole. Matlab, with time, these fibers will also grow na, because they have to push them to the opposite pole. Let's say, just imagine it like this. It is growing. It is growing. So basically the agenda is to send these centrosome to the opposite poles, and obviously to do that, astral rays are specifically helping in this process only. Astral rays are specifically helping in this process only. Plus further some continuous, discontinuous fibers will also form. Right? That type of structure is going to form. That type of structure is going to form. Are you getting my point? That type of structure is going to form. Are you getting my point? Right? So centriole, the centrosome is going to help here. Okay? So it is in the animal cell, that's why. And the ester formation is there, astral rays form here. That's why we use the word amphiastral. Amphiastral mitosis in the case of animals. But in the plant cell, we do not have that. Right? In the plant cell, most of the time students, they ask me this question. I don't know why you people are not asking that in the plant cell, if there is no centriole, then how do the spindle apparatus will form? In the last class, when I was talking about the microtubules, I told you that microtubules are very important for the spindle fiber formation. If you remember this thing, if you know this thing even a bit, right, you know na, your chromosomes will be arranged, right? The chromosomes, yes, if you remember, if you know that chromosomes, they'll be arranged, that's how they will be fixed, something like that. Yes. Right, so these short astral rays are giving it aster-like appearance. So you know the spindle apparatus formation, these are the spindle fibers, right? We need them. Why? For the separation of these chromosomes, right? Right. So basically, now if in the plant cell we do not have centriole, then what's going to happen? See, imagine, in the plant cells we do not have the centriole, but bache, pericentriolar material is there. What is there? But pericentriolar material is still present. Let me tell you this. When you talk about these centrosomes, they are sometimes also referred to as MTOC. Right? They are also referred to as MTOC. Do you know the full form of MTOC? Yes? Do you know the full form of MTOC? Anybody in the class? What is MTOC? This is the word that I have used. Centrosomes are MTOC. Yes, bache, they are the microtubule organizing centers. Right? They'll organize these microtubules, because these microtubules are required for the formation of the spindle apparatus. So they are MTOC. Yes, bache. Mr. Tilu, please focus here. Right? Let's not talk about the Instagram. Please focus here. Okay? So MTOC, centrosomes are a microtubule organizing center overall. That's what we say. That's why the tubulin protein will polymerize and they are going to form the fibers like this. But the point is actually it is the pericentriolar material, right, that helps in the polymerization of that tubulin fibers, right, that mainly helping in the this pericentriolar material. Actually it is the pericentriolar material that is helping in the polymerization, or you can say that the formation of that spindle fibers. Okay, this pericentriolar material actually helps in the formation, or you can say that polymerization of that fibers, or formation of that spindle apparatus or spindle fibers, right? And what is the role of these centrioles? Centrioles will provide it the orientation, it will give it a proper orientation, right? But mainly when it comes to the polymerization and the formation, this pericentriolar material is involved. So now in the plant cell we do not have the centriole. So obviously when it comes to the formation of aster rays, right? So aster rays are formed by the centriole only. So no astral rays will form, plus it is just taking care of the orientation. So in the plant cell also spindle fiber will form, but it will not be properly orientated. It is not properly orientated. Are you getting my point? It is not properly orientated. Stella, is that clear? Is that clear, guys? Please focus. Please focus. Done? Okay, so that's why amphiestral and anestral word is used. Now obviously I directly start the directly start the discussion from this part, not from the prophase ki this is going to happen in prophase, that is going to happen in prophase, because now in one flow we will go, in just a flow we will go. Okay, but before that you should know what these structures are, what are centrosome, what is their role. So it is MTOC, microtubule organizing center. It has to polymerize the tubulin fibers. It has to form that spindle fibers. Some are continuous, some are discontinuous. In the spindle apparatus you will see that different types of fibers, right? Some they will attach to the chromosome, they have to separate the chromosome. So that's what we have to discuss. So when you talk about the nuclear division, this mitosis, let's say the cell division has started. Compare it with your NEET examination, guys. Just compare it with your NEET examination, guys. When you will start attempting that paper, when you are inside the examination center, they will not give you the books to read the questions, isn't it? They are not going to give you the books to read the questions. Am I right? Am I right? They are not going to give you the books. Okay, you can read something on. No. Now whatever you guys have done, you just need to right, execute the things now, right? Whatever you have learned so far, you have to execute that. That's the story. That's the thing about Arpana, right? Arpana, that is what you have to do. So same is the case here. When the mitosis will start, right, when the cellular division will start, there is no further protein synthesis, right? There is no further protein synthesis. Already all proteins are available there. Already the stock of energy is there. So nothing like that is going to happen. It's just that. And whatever, some cytoskeletal element, these microtubules, those who are present there, they will even depolymerize. They will even depolymerize when there is when that division will start, especially in the animal cell. So what's going to happen? Your animal cell will somewhat become rounded. Its cytoplasm will become viscous, because these microtubules, they will depolymerize. The tubulin will be there in the cytoplasm, right? And it will be used Later. It will be used later. So your centrosome, right, your pericentriolar material, it will polymerize it. It has to form that spindle fibers. Are you getting my point? Right? So in the animal cell, because centriole is there, then ester-like, star-like appearance is there. That short fibers will radiate from that centriole part. Okay? And that thing will not be there in the animal cell. That thing will not be there in the animal cell. Is that in the plant cell? Sorry. Okay? Done? Done? Okay. Is that clear? Is that clear? Okay, so that's how it is. So let's start the discussion there in detail. Ultimately, we have to revise it from the NCERT, but you should know what is happening in early prophase, in the middle prophase, and in the late prophase. So whenever you are talking about the prophase, whether it is the prophase of your mitosis or the meiosis, just keep one thing in your mind. Just keep one thing in your mind, and that one thing is that ultimately condensation of chromatin occurs. Condensation of chromatin occurs. Condensation of chromatin occurs, and that will form chromosome. That compact, thick structure. Okay? Ultimately, that is going to happen in the prophase. Right? In the case of meiosis, because it is a reductional division, so pairing of homologous chromosome, this crossing over and all, we have to discuss it there, but it is the mitosis only. It is the mitosis only. So ultimately in the prophase, what's going to happen, students? The condensation of the chromatin material will be there. That will result in the formation of the chromosome. That will result in the formation of the chromosome. So please keep it in your mind. Please keep it in your mind that ultimately this thing is going to happen. So now one by one, I'm going to explain it to you. So one thing that you have to remember, right bache, that when this mitosis will start, right, when this mitosis will start, right bache, what is going to happen? What is going to happen? Yes, what is going to happen? Tell me. A lot of things will happen na. Chalo, let's start. And yes, how can I forget? This term was proposed by Fleming. This term was proposed by Fleming, term mitosis. Term mitosis was proposed by Fleming, and Fleming was the one who observed it in animal cell. I'm being very specific, right? Who observed it in animal cell. ठीक है जी, Fleming. Okay, so he termed it mitosis. He proposed this term mitosis. Right, bache? Right, bache? So in animal cell, he basically, so in animal cell, he described this mitosis. Is that clear? Chaliye. So we are talking about the this prophase. Okay, and we will start with the early prophase first. Okay, I'm not going to write it like this, ki early, late, early, middle or late, but ठीक है, initially what is going to happen, right? So initially what is going to happen, let's first talk about that, right? So as I said, bache, as I said, what is going to happen there? There will be the depolymerization. Depolymerization of cytoskeletal element. Right? There will be the depolymerization of what? Of the cytoskeletal element. Yeah, you can say that the cytoskeletal microtubules. Yeah, simple skeletal microtubules. Let's be more specific. Okay, your microtubules. Done, bache? Right. And they will be depolymerized into their protein subunit. So into their protein subunit means into tubulin, right? It is the tubulin that is making it now. So it will be depolymerized. So gene expression, even the protein synthesis, everything will stop when this prophase will start, right? When this prophase will start. So gene expression, yes, protein synthesis, right? All of it will stop. And even cell motility. will also stop, because now it is dividing and it will not be able to secrete anything. It will not be able to secrete anything. Now obviously you tell me, if bache there is a depolymerization of the microtubule, right, so obviously that will turn cytoplasm, that will make cytoplasm viscous, because that tubulin will be there in that cytoplasm. Now cytoplasm will become viscous. So these kind of changes will occur. These kind of changes are going to be there. And let me tell you, even the organelles, they will also disappear in the prophase. The cell organelles which are present, they will also disappear, right bache? They will also disappear in this prophase. Okay, they will also disappear, right bache? They will also disappear. Just say, let's say your endoplasmic reticulum, your Golgi apparatus, they all will break into small vesicles. They all will break into small vesicles. Are you getting my point? Yes, right? Are you getting my point? Now that part will come. Just say I told you that your centrosome, your centrioles, they are firmly attached to the nuclear envelope. They are firmly attached to nuclear envelope. Am I right? They are firmly attached to nuclear envelope, right? These centrioles. Now they are duplicated. We already know they are already duplicated, right, in the S phase. Now bache, because they are firmly attached to the nucleus, they need to move to the opposite pole, right? They need to, yeah, they'll move to opposite pole. And how is it possible? I just told you, right? We have already covered that part. Let's say this is your cell, right? Yeah, it's something like this. They are too close to each other, right? This is the nucleus part here, this is your cell here. So if it is the animal cell, that small fibers will radiate and they are going to just move it towards the opposite pole. Is that clear? They are going to move it to the opposite pole. Is that clear? Is that clear? So some short radiating fibers, the microtubules basically, they will, right bache? Right? Yes? Right? चलिए, so now to be very specific here, जैसे ये देखो, what's going to happen? Obviously these are the short radiating fibers. These short radiating fibers, how will they form? Actually what's going to happen in this early prophase? Your bachche tubulin protein, your tubulin subunits assemble near centrosome. They'll come near centrosome and then they'll be polymerized. Okay, then they will be polymerized. Okay, ultimately though they are microtubules only, so they are forming some short radiating fibers, right? They are forming what? They are forming some short radiating fibers, and these fibers are the astral rays. These fibers are the astral rays. Now you see here you have the centriole, it is covered with the pericentriolar material. So just let's say that's how these fibers are coming out. These astral rays are coming out. So it is giving it a star-like appearance. That's why, because astral means star, and these are the astral rays. So these astral rays are not directly radiating. It's not like that these astral rays, they are radiating, right? It is literally not like that these astral rays are radiating from the centriole. No, you will see it like this. Just these are the two centrioles at 90 degrees to each other, right? Diplosomes basically covered with the pericentriolar material, right? Or you can say that as some amorphous substance, just or you can say that as some kind of, cytoplasm. A kind of like it will appear like this. It is not like that it is directly coming out from the centriole. No, not at all, right? It is not in the direct contact with the centriole, right bache? Right bache? So they are basically separated. These astral rays, they are separated from this centriole, right? they are separated from centriole by a zone of cytoplasm, right? Zone of cytoplasm, they are separated. Is that clear? Now it is looking like a star, so as amphiestral mitosis. Now these fibers, right, they are going to push these centrioles to the opposite pole. Then we will come to the status of that chromatin also, ki they are undergoing compaction or not, but firstly understand this, right? These short fibers, they need to move each other to the opposite pole. So obviously when they are moving each other to the opposite pole, so obviously in between, don't you think that they will grow? They will also grow. Yes, don't you think so? Don't you think so? Yes. जैसे as the centrioles move apart, the microtubules stretching in between, they grow in length, है ना? Obviously they will grow in length because they are moving each other to the opposite pole. So they are also going to grow in length. Am I right? Am I right? Yes, bache. Am I right? So basically microtubules stretching between them will increase, right? Right, bache? And they will increase in length, right? They will increase in length. They will increase in length and even in number. Their number and length will increase. Am I right here? Their number and length will increase. Am I right here? So astral rays are mainly helping in moving the centriole apart, right? But here, right, we need to discuss it further, right? So basically microtubules will stretching between them. They will increase it in number and the length, right, bache? So astral rays are going to shift the centriole to the opposite pole, isn't it? Isn't it? Yes or no? Yes or no? Right? Right? So ultimately these astral rays are helping the centriole to move to the opposite pole, right? And then further the spindle fibers will form. So remember, for spindle fiber formation, we do not need that aster rays properly. We do not need that centriole. That pericentriolar material can do this thing. Right, that pericentriolar material can do this thing. And even I will tell you one point related to the kinetochore as well. Right, I will tell you one point related to the kinetochore as well, but let me explain this. So are you able to understand that? So aster rays are basically helping these centriole to move to the opposite pole, right? And due to which the microtubule stretching is also going on in between, that will result in increase in the length and the number of these microtubules here, number one. Secondly, the spindle fibers will also form. Basically overall in spindle apparatus in animal cell, you will see astral rays, you will see spindle fibers, you will see some continuous fibers, right? You will see some discontinuous fiber. They need to attach to the chromosome, they need to separate it. Clear bache? Clear bache? Are you getting my point? Are you getting my point? So for spindle formation, always remember. And why do we use the word spindle? Because this spindle apparatus, its shape is also like this. Spindle means it is tapered. It is tapered at ends. It is tapering at ends and wide in middle. Wide in middle. That's why we use the word spindle here. Right. So ultimately right. So spindle fibers will form, and for that pericentriolar material will help in its formation. So microtubules are required. Microtubules are required. So any doubt here? Yes, please. Any doubt here? Yes, please. Yes. So centrosomes are the microtubule organizing centers. Is that clear? Is that clear? Sure. Right, so microtubules, they'll be arranged in bundle. They are forming the spindle fiber, which are much needed to separate the chromosomes. Okay. Okay, so this is like this. So now here in the center, the central part of the spindle apparatus, the central region here, it is actually the center of the cell. Also you can say it is the equator. It is the equator. It is the equator. We should know that. Chalo, let me show you the picture. Ye dekho. See this. Prophase. Right? These centrioles are moving to the opposite pole. Can you see bipolar spindle is forming in between? These are the ester rays, and slowly we will see some changes will also be there in the nucleus. Right? Some changes will also be there in the nucleus. Just have a look. Aur ye dekho. These are the stages of cells division under the microscope. Right? These are the stages of cell division under the microscope. And trust me, you have to understand the mitosis first. Matlab prophase mainly. Rest stages are easy. Then we'll move to meiosis. Meiosis it is the prophase one, which is very long and which is little difficult. Matlab not exactly difficult, but yeah, you might consider it complicated. Then everything will be easy. So mainly we need to put our energy to understand what prophase is. Okay, so we can finish this chapter. I'm not like that, I will explain it. I'm not going to repeat the same things. You understand mitosis properly, you understand prophase 1 properly, trust me, rest everything is easy like this. That's the beauty of this chapter. So ye dekho, these are the different stages, right? We used to do this in school or even in the college. Jaise ye dekho, chromosomes are so clearly visible here. Ye dekho, they are at the opposite pole. It is showing the anaphase here. Ye dekho. Okay, they are arranged at the equator here. All the chromosome. It is metaphase. It is metaphase. ठीक है? Okay. Now, what's the condition? And we are still in early prophase, okay? Now, what is happening to the nucleus? Dekho, chromatin threads are there, bache. They'll start condensation. Their compaction will start, their condensation will start. They'll start losing water, right? Matlab dehydration, literally dehydration will also occur here. Do you know that? Right? So chromosomes, right, when it was in the interphase also, it was just the chromatin threads, no? They were just the chromatin threads, na? So chromosome, initially they will appear like long thread-like structures, right? Thread-like structures. Initially they appear like this. Now you tell me here, chromosomes are in monad state or in dyad state? They are in monad state or in dyad state? Yes, please. Tell me. Aditya, kindly focus here. So they are in monad state or in dyad state, the chromosomes here. And overall in the nucleus, the amount of DNA is 2C or 4C? Please tell me. Is it 2C or is it 4C? Yes? Bache, obviously this is the nucleus, right? Right. Daughter cells are not formed yet. So obviously it is 4C. The DNA amount here is 4C, but the number of chromosomes will remain same. Those who are writing 2C, please watch the lecture again. Right? Please watch the lecture again. So initially chromosomes are long, they are thread-like. Right? Mitosis. When we are using the word mitosis, na, mitosis is also having the same meaning. Right? Mitosis is also having the same meaning. Matlab thread state. Mitose means, right, thread-like state. Thread-like state. That's why, so chromosome initially they look like thread. Right, bache? Right. So chromosomes, right, here they overlap. Right, they have their ends are invisible. Their ends are invisible. I'm talking about the initial stage yet. Ends are invisible. It looks like a ball of yarn. Okay, it just looks like a ball of yarn, or in simple words I can say they are tangled. These threads, they are tangled, right? They are tangled. They don't have the initially in the early prophase, chromosomes are not having the discrete entity that I'm chromosome 1, I'm 2, I'm 3, I'm 4, I'm like this. No, No, not at all. Not at all. It's just like the ball of yarn, right? The ends are not clear. Chromosomes are overlapped over each other. They are invisible. Ends are kind of invisible. So they are, right? It is looking like the ball of yarn. Right, bache? Is that clear? The invisible ends look like a loose ball of yarn. Right, bache? Right. Now gradually, matlab jaise, let's say initially early prophase, that's the state. So gradually what's going to happen? Gradually means slowly. Chromosomes, they'll become thick. If they are becoming thick and the condensation is going on, right? The condensation is going on. There will be the loss of water from that chromatin. Condensation will occur. They are becoming thick. And obviously even they will become short also. And as I said, the chromosomes here are in di-8 state. Why, bache? Because 4C amount of DNA is there. 4C amount of DNA is there, right? After S phase, the amount of DNA get doubled. So here, remember what's happening. Chromosomes, gradually chromosomes, they will become short, they'll become thick. Why? The condensation is there, right? The loss of water is taking place. Is that clear? Is that clear? So along with this loss of water, there is the coiling of chromosome as well, right? In the molecular basis of inheritance also we discussed now that further the chromosome, the chromatin, they will coil. They say firstly histone proteins, then some non-histone proteins will also take the part. So basically solenoid model, and then further loops will form. I don't know whether you know it or not, but one thing that you can remember, just say, let's say, if it is something like that, then slowly it's coiling, its condensation will occur. So definitely that is going to make it short as well. Are you getting my point? So ultimately in prophase, what is happening, bache? Right, the chromosomes condensation will be there. The condensation of chromosome will be there. Is that clear? Is that clear? Sure. So obviously the chromosomes that we are going to see here, they will be like this. Right, diade, right, two sister chromatids, they are attached to the centromere. They are attached to the centromere. Done, bache? Done? Yes? Done? Sure? Are you sure? So chromosome will further shorten, they will thicken, their chromatids will also kind of uncoil, right? So finally after that, after further compaction and all, they will acquire this shape. And you know that when you talk about the kinetochore, from both the sides here, they are covered with a disc shaped protein, right? They are covered with a disc shaped protein, that is kinetochore. Right, and remember one thing, bache, the spindle fibers, they always attach to the kinetochore. They never directly attach to the centromere. That is important. Right, that is important. Right, and this kinetochore, I don't know whether you know it or not. It serves as a, I told you now, I'll tell you a point regarding this kinetochore. So it serves as a center of nucleation of tubulin proteins, right, bache? So it is the kinetochore, the disc shaped protein which is covering the centromere from both the sides. So right, bache? Yes. So spindle fibers, they'll attach to this. Okay, they will attach to this. Done? Yes. So it is a nucleation center for tubulins, for tubulin polymerization, right? This kinetochore, remember, this kinetochore is also the nucleation center for the tubulin polymerization. Is that clear? So basically it is happening in the early prophase. Middle prophase may be chromosome will further thicken, they will shorten, right? Now they will acquire their proper shape, right? That characteristic shape, right? And moreover, what is happening, bache, in that middle prophase? Even your nuclear lamina will also disintegrate. Its disintegration will also start, and by the end of prophase, it will completely be degraded, disintegrated. And even, you know, nucleolus is there. Nucleolus, that is the ribosome factory. Do you know about nucleolus, which is the ribosome factory, right? So in the nucleus, more than one nucleolus can be there. So that's why I'm using the word nucleoli, right? That is why I'm using the word nucleoli here. They also start decreasing in their size in the middle prophase, and finally they will disappear. Is that clear? Is that clear? Yes, bache. So nucleoli, they start decreasing in size. And finally disappear, and their, you know, their nucleic acids will go to some chromosome or something. It is like that, right? And obviously all the organelles, they will also disappear. Okay, bache? So spindle will get proper shape, nuclear envelope will break, the chromosomes will be there in the cytoplasm, right, bache? Yes, right? So by the end, you can say that in the late prophase or by the end of prophase, what's going to happen? Yes, I'm writing the points here, they are important. So spindle, that spindle assume proper shape and size. Right? Then next is bache, nuclear envelope breaks down fully, and chromosomes, nuclear envelopes, they break down fully, and chromosomes, they'll be released in the cytoplasm. And you know, even the central cytoplasm, it will become, it was initially viscous, no, it will become fluid like, so that chromosomes can move freely towards equator. Is that clear? They can move freely towards equator, right? So spindle will assume proper shape and size, nuclear envelope break down fully, right? Central cytoplasm will become fluid like, right, bache? Right. Yes. Is that clear? Is that clear? Right? So that will happen by the end of prophase, and the complete and now it is PMAT. So after prophase, what will be there? There will be metaphase. So in this particular sub stage, what is going to happen? The full condensation of chromosome will be done. Right, bache? Is that clear? So this is the prophase. So ultimately, nuclear envelope will disintegrate, bache. Nuclear envelope will disintegrate, and the chromosomal compact chromosomes will be there in the cytoplasm. Now the cytoplasm is also fluid-like. So the chromosomes, they'll come to the equator. Matlab equator means center of the cell, and it is important. That is what we are going to see in the metaphase. Vinay, Crystal, ma'am, not clear. Chaliye, tell me, bache. I'll try my best to make it clear to you. So see the diagrams, right? Obviously, we have to follow the NCERT. Diagrams are not so good, but yeah. Early prophase, this is of late prophase. And this one is the transition to metaphase. It is not the metaphase, transition to metaphase. So see, this spindle fibers are attached to the kinetochore, right? And finally these chromosomes, they'll come to the center. They'll align itself here. Okay, so diagrams are important. Chaliye, so let's cover it from the NCERT and then let's go. So when it is the prophase, which is the first stage of karyokinesis, right? It follows the S and G2 phase of the interphase. Simple it is, right? In the S and G2 phases, the new DNA molecules formed are not distinct, they are not separated, right? You cannot say that, acha, this is this arm of this chromosome, yeah, it is chromosome 1, it is chromosome 10. No, it is not distinct, but intertwined, right? Jaise loose ball of yarn, you can imagine. Ki we cannot say, acha, this is that end of chromosome or something. No. So in the prophase, marked by initiation of condensation of chromosomal material. This chromosomal material will condense. Further coiling of that chromatin material will be there, right? So chromosomal material becomes untangled during the process of condensation, right? Because it has to get that discrete identity. So centrosome which had undergone duplication during S phase, they will start moving towards the opposite pole of the cell, right? So these are the characteristic event that we need to remember. Okay, जैसे chromosomal material condensed to form compact mitotic chromosomes. So they are seen to be composed of two chromatids attached together at the centromere. Okay, diet state is there. So centrosome which had undergone duplication, right? The duplicated centrosome, they start moving towards the opposite pole. Bache, each centrosome radiate out microtubules called esters, and it is there in the case of animal cell. So two ester together with spindle fibers form mitotic apparatus. Yes, obviously you need to remember this. Any doubt? Any doubt? Very good. So cells at the end of prophase, when viewed under microscope, do not show Golgi complex, ER, nucleolus and the nuclear envelope. Very important point it is. Okay? Very important point it is. Chaliye. Next is bache metaphase. You know, this stage is also very interesting. Anybody in the class who's aware of this thing, karyotype? Do you know karyotype? Anybody? That karyotypes, ideograms, we can obtain in the metaphase stage. So anybody in the class who knows what karyotype is? Anybody? Yes? Right? Karyotype. But it is a kind of visual presentation of our chromosome. It's a kind of visual presentation of our chromosomes, right? Of an individual's chromosome, its form, its shape, its size, right? They'll be seen, right? They'll be seen. This karyotype will show us that it is a visual presentation of our chromosome. So it is used to check if there is any chromosomal abnormality. Jaise when you talk about the Down syndrome, right? Bache, that is trisomy 21. That is trisomy 21 means there is one extra copy of chromosome number 1. So if a person is having Down syndrome, that is trisomy 21, that person is having 47 chromosome because they have one extra copy of chromosome number 21, and that with the help of this karyotype we can see that. Right? So we can obtain it, right? Because metaphase is the best phase to get that karyotypes and ideograms. Okay, so now you know that the spindle fibers, they will attach to what, bache? Always remember they are attached to the kinetochores. They are attached to the kinetochores. They never attach directly to the centromere. So jaise let's say, again take the example of a cell having 4 chromosomes. So they all are going to align itself at the equator, right? Equator. And these spindle fibers are attached to what? They are attached to the kinetochore. This is what metaphase is. That's it, that's all. This is what metaphase is. So all the chromosome, they will align itself at the they are aligning themselves at the equator. So this is also known as metaphasic plate, right? Metaphasic plate. This is what metaphase is. This is what metaphase is. Let's say we are taking the example of a cell having 4 chromosomes. So that is how they will be arranged. Actually, it is the metaphase. Best phase to study the chromosome shape, size, their form. Best phase. Now, after metaphase, there is anaphase. But anaphase mein na, we have to. These chromosomes, they will start moving to the opposite pole. Ye dekho, that's your chromosome. That's your chromosome. We have to move it to the opposite pole in anaphase. So in anaphase na, we have to split it. If I want to move it to the opposite pole, I have to split this. I have to split it. So there will be the splitting of centromere. You imagine the diagram. Splitting of centromere and separation of sister chromatids will be there. Splitting of centromere and separation of sister chromatids. Splitting of centromere and separation of sister chromatids will be there. So basically what is happening in your anaphase? In the anaphase, the depolymerization of these tubules will occur, right? The depolymerization of these tubules will occur, and due to which there will be the splitting. There will be the splitting of your centromere and these chromatids, right? Because see, you are going to split this chromosome. Matlab it's like that ki you are having a chocolate, you are dividing it into equal halves. One you will give to your brother, or other part you will keep. You are going to keep it na ki yaar, I need it, I want it. Same is the story here. Same is the story here. Okay? This is the chromosome. We are going to split the centromere, and there will be the separation of the sister chromatids. In the anaphase, that's going to happen. In the anaphase, actually, there is the depolymerization of these microtubules. That is why, due to which, na, it is just like ki something is pulling them towards the opposite pole. It feels like that. It feels like that. Right? It feels like that. So, the centromere will split. These sister chromatids will start moving towards the opposite pole. Now they are not like sister chromatids, bacha. Now they are not like sister chromatids. Now they will start behaving like chromosomes, na. They are separated. When these two arms, they are attached to the same centromere, you are calling them as sister chromatids. Now you are splitting it. Now you are splitting it. Just imagine like this. Now these two sister chromatids will start behaving like the chromosome, right? They will start behaving like the chromosome. So it is the anaphase of mitosis. It is the anaphase of mitosis. Right now, just I told you here, obviously the kinetochore is there, and I told you it is a nucleation center for the tubulin polymerization. Now in anaphase, depolymerization is there, and that's why it is moving to the opposite pole. Arms will trail behind like this. Just imagine this is the centromere and the kinetochore. Arms will trail behind like this. Just imagine. That's how it will move. Are you getting my point? Just a depolymer, microtubules are attached to it, spindle fibers are attached to it. So now their depolymerization is occurring, so it will behave like this. So arms are trailing behind, right? It's like, just let's say there is a kid who's basically crying, right? Mama, I need that toy. Mama, I need that toy. These days, so mamas are like, oh baby, take it. But when we used to go to the market, right? And if we start behaving like that, so our mother used to hold us, right, from our waist and like, chalo, let's go. And we are like, I want that toy. Same feeling. So arms will trail behind. Arms will trail behind. Okay, arms will trail behind. So obviously, if it is a metacentric chromosome having equal arms, then you will see V-shaped chromosome. If it will be submetacentric, you will see kind of L-shaped chromosome, isn't it? Isn't it? If it is submetacentric, what will you see? You will see S-shaped. You will see S-shaped. Yes, S-shaped chromosome. Sorry, V and then L-shaped. L-shaped, my bad. L-shaped. And then acrocentric and telocentric is also there, na? Then L-shaped. Right? So there will be different shapes of chromosome, and obviously it depends upon what? Right? On the size of these arms. It's like that. So this is going to happen in anaphase, and for moving each chromosome to the opposite pole, it takes 30 ATP. For this each chromosome, if we want to move them to the opposite pole, it takes 30 ATP to do that. 30 ATP. So imagine the number of ATPs, yeah, energy that we need. Imagine. Just imagine. I'm talking about each chromosome. Okay? Right? Chaliye. So let's revise it, and then let's move to the telophase. Telophase is just opposite to the prophase. Whatever happened in prophase, opposite to that will be in telophase. Now obviously in anaphase, your chromatin material, right, because they are getting separated, they'll reach to the opposite pole. So in telophase again, these chromosomes, now they will not remain discrete. They'll lose their identity. Again, they will become thread-like. Again, they will become tangled. Again, the nuclear envelope form. Again, the organelles form. Again, the nucleolus will appear. Right? And obviously, these centrioles, these centrosomes, they'll be distributed to the daughter nuclei like that. That's the story. That's the point. Okay, so complete disintegration of nuclear envelope, मतलब totally disintegrated. The nuclear envelope will be this complete disintegration of nuclear envelope is marking that now it's the time for the metaphase. So chromosomes, they'll spread through the cytoplasm. So condensation by this stage is complete. Matlab by metaphase, the condensation is done and dusted. Right? We can clearly see the chromosome under the microscope. Right? So morphology of chromosome is most easily studied here. It's a previous year question. It is a previous year question. Right? So metaphase chromosome is made up of two sister chromatids held together by centromere. Disc-shaped structure kinetochores are present at the surface of centromere. So these kinetochores are the site of attachment of spindle fiber. So just here they have mentioned formed by spindle fiber. You can say formed by microtubule or something. Yes, simply you remember that kinetochores, they are the nucleation center for the tubulin polymerization bus. Right? Right? Okay. So these structures serve as a site of attachment of spindle fiber to the chromosome that are moved into the position at the center of the cell. So metaphase mein chromosome will lie at equator bachche, right? So one chromatid of each chromosome is connected by its kinetochore to spindle fiber, right? From one side pole, and its sister chromatid is connected to the other side, right? So this plane is the metaphasic plate. So these are the two characteristic feature, these are the two things that we will see in metaphase, and this is the diagram. So dekho, they are arranged like this. Okay, so spindle fiber attached to the kinetochore and chromosomes are moved to the spindle equator and get aligned along metaphase plate through spindle fibers. Any doubt? Any doubt? Hanji, jaldi batao. Any doubt? Chaliye, next, Mr. Anaphase, look at the image. So at the onset of Anaphase, each chromosomes arranged at metaphase is split simultaneously, and two daughter chromatids, which are now referred to as daughter chromosomes of the future daughter nuclei, will start migrating to the opposite pole. That's the beauty of this. Okay? So chromosomes move away from the equatorial plate, and centromere of each chromosome remains directed towards the pole, and arms are trailing behind. Vinayak bache, arms are trailing behind. Right bache? So centromere will split, chromatid will separate, chromatid move to the opposite pole. Doubt? Doubt? Hanji, doubt? I told you na that you have to put your energy to understand what what do we say? Prophases. Rest everything is simple. It is very simple. Yes? So if we need to study the morphology of chromosome, it is the metaphase. Then anaphase में splitting will be there. है ना? ठीक है. Any doubt? Yes? Any doubt? Okay. One second. ठीक है, done बच्चे. So now chromosome will move to the opposite pole. So these daughter या sister chromatids are now known as chromosome. It's like that. So देखो, in the prophase, right? In the prophase there is the compaction of the chromosome, right? In the prophase there is the compaction of the chromosome. Yes or no? in the prophase there is the compaction of the chromosome, obviously. And why that compaction is required? Because in the anaphase then it will be easy to move that compact chromosome to the opposite pole. So see, everything is associated with each other. So here depolymerization of the microtubules will occur at that kinetochore end, energy will be used, and that's how they will move. So now telophase is also very, it is also a kind of complex process. It will take time. Anaphase can be it can last for few minutes, but this telophase is going to take time. Okay, so at the beginning of the final stage of karyokinesis, right, the chromosomes, they have reached their respective pole, they will again decondense. In the prophase, they condensed. Here, they decondense. Again, they will lose their individuality. They will not be as a separate unit. Right, now they will become tangled. Right, bache? So, individual chromosome no longer be seen. Each set of chromatin material tend to collect at each of the two poles. Right, so chromosome cluster at opposite spindle poles. Their identity is lost as discrete element. Nuclear envelope will again form, bache, right around the chromosome cluster at each pole. Right, then there will be the two daughter nuclei, nucleolus, Golgi complex, ER, they all will reform. Is that clear? So that's why we say it is just opposite to the prophase. Okay, chaliye. Next is cytokinesis, that is cytoplasm division. Sometimes it is also referred to as D phase, right? D phase. But it is different in animal and plant cell, right? It is different in animal and plant cell. Now, let's say if this karyokinesis, nuclear division, if it is not followed by the cytokinesis, then we will get a multinucleated condition that is known as cenocytic condition. Cenocyte it is, right? Cenocytic condition means multinucleated condition. Multinucleated condition, right? But sometimes we also use the word syncytium. Syncytium is also the multinucleated condition, but syn means fused. So sometimes some cells, right? Some cells. Sometimes what happen? The cells, right, their common walls, they disintegrate. Their common walls, they disintegrate. It's a multicellular structure, but the common walls, they disintegrate, and it results in the formation of a multinucleated cell. So it is the syncytium. It is the syncytium. Are you getting my point? It is the syncytium. Hope it is clear. Now cytokinesis, cytoplasmic division, it is different in animal cell and plant cell. Why? Because in plant cell there is a cell wall, but in animal cell there is nothing like that. So in the animal cell, the thing that you need to remember is that there is the formation of a contractile ring. There is the formation of a contractile ring. But the word is contractile ring. Definitely you are talking about the actin here. You are talking about the actin here. Right? So that is how the cytoskeletal elements are useful. Even in the last class I told you. So from periphery towards the center. Right? This contractile ring, it is forming a kind of this. There is a cleavage furrow that will form. That cleavage furrow is basically having the actin. So from periphery towards center, okay, this cleavage furrow will go. From periphery towards the center, the division will occur. Okay, so that's why we are saying centripetal. Right? Here the cytokinesis is centripetal kind. So, ये देखो contractile ring will form. ठीक है जी. So in the animal cytokinesis, cleavage furrow will form. Right? So right, बच्चे. So cleavage will process centripetally. Is that clear? Is that clear? Yes. Is that clear? Done? Okay. So in the animal cell, this cytokinesis, it starts. Right? It starts. Obviously, when? It is going to start. This animal cell mein cytokinesis, when will it start? When will it start? After telophase, well, after anaphase, when the cell is in early telophase. Right, and by the end of telophase it will be completed because cleavage furrow will form. No cell, because here we do not have the cell wall, no cell plate formation, nothing at all. The cleavage furrow will form, that is the contractile ring that is having the actin, right? And centripetally this cleavage will proceed. Centripetally this cleavage will proceed. I'll give you the one more example. ये देखो, from periphery towards the center. So it will start in early telophase. By the end of telophase, it will get completed. Now, in the plant cell, this cytokinesis is different. Actually, it starts by the end of anaphase. Imagine, it will start by the end of anaphase. So actually here what is going to happen? Because we know that in the plant cell wall is there. So here you will see the cell plate will form. Right? This cell plate will form. And let me tell you students, this cell plate will further form that middle lamella. We discuss about the middle lamella, na? The cell plate is going to form that. And see, it will first form in the center. So here it will move from center towards periphery. From center towards periphery. Right, bache? So it is centrifugal. Okay, and here you will talk about the phragmoplasts as well. What are we going to discuss? We will talk about the phragmoplasts. Okay, bache? So in plant cell, what's going to happen? Right, by the late anaphase, this will start. Right, bache? This cytokinesis will start. Okay, okay, yes. Right? Chaliye, let's discuss that now. One minute. Chaliye. So, how will it form? You remember, I told you that Golgi apparatus help in the cell wall formation, if you people remember, right? Golgi apparatus, they help in the cell wall formation. So basically, as I said, it will start by the end of anaphase or sometimes in early telophase. And sometimes we say that animal cell, this cytokinesis, matlab it will just initiate in the late telophase, in the telophase itself, and by the end of telophase, it will get completed. Now, here the point is, they say your Golgi apparatus, your small Golgi vesicles, or sometimes endoplasmic reticulum, they will come here in the center. They'll come here in the center, and then they will just, you can say that they will kind of fuse. That vesicles will come here in the center, they will fuse, and they form the cell plate. Okay, and that cell plate will grow from center towards the periphery. It is moving from center towards the periphery. So centrifugal is the word. Secondly, in the plant cell, this spindle apparatus will not degenerate. Just even in the animal cell it will degenerate, na? But in the plant cell it is not going to degenerate early, right? So this spindle apparatus plus this cell plate, right? We call this structure as phragmoplast. What's the word? Phragmoplast. That's the word. What is the word that we use here, bache? What is the word that we use here? It is phragmoplast. Yes, please write down. It is phragmoplast. Okay, phragmoplast. Right? So even you will see it during late cytokinesis. Okay? Right? So it is having microtubules. Okay. Phragmoplast. Right? So it will be there in the plant cell only. And it will form during late cytokinesis. It will form during late cytokinesis. Let me explain this. Yes, done, bache? right? So in the phragmoplast, wait one second, phragmoplast is the word that we use in the case of plant cell. मतलब plants में obviously it is different. जैसे your Golgi apparatus or endoplasmic reticulum, their vesicles will come here, they will fuse. Now what is going to happen? जैसे you can say some microtubules, some microfilaments, yeah, you can say in late cytokinesis na some structures will form here, right? These cytoskeletal elements are going to form a kind of structure here. Matlab it's a kind of spindle apparatus. It's a kind of spindle apparatus which will form here. It is a kind of spindle apparatus which will form here in between two nuclei. The nuclear envelope is reforming. So actually it is important. It is just like a spot where, you know, this basically you can see the cell plate can form and can grow here, right? Cell plate can form, can grow here. It is like that. Okay, it is like that. This is the phragmoplast. Okay, so at the center of the cell the vesicles will fuse, right? The phragmoplast will form, right bache? Yes, so this cell plate will form middle lamella. It represents middle lamella. Okay, it represents middle lamella. Okay, done. So this phragmoplast helps in the formation of your cell plate. Okay, right. So it is having microtubules, it is even having the actin filament. Done. Right, so it is acting like a scaffold for the cell, cell plate synthesis. Okay. Okay. Right, so basically this phragmoplast is having microtubule, microfilament. It is acting like a scaffold for this cell plate. That's it. So ye dekho, in animal cell microfilament ring. So you know that microfilament and actin is same. Okay, microfilament and actin is same. So that's how this cleavage furrow will form. Ye dekho, this is the cleavage furrow. It is growing centripetally, and here this is the cell plate. So here somewhere here you will see that microtubules and microfilaments will also form a structure, and we are calling it as what? We are calling it as phragmoplast, right? So it is moving from center towards the periphery. So it will represent middle lamella. It is going to represent middle lamella. That's it. Chaliye, so let's cover it from this side. So mitosis accomplished not only the segregation, separation of duplicated chromosome into daughter nuclei, but the cell itself is divided into two daughter cells, right? So in animal cells, see, the furrow is there, and that is made up of your microfilament. So furrow will gradually deepen and ultimately join in the center, dividing the cell cytoplasm into two. Okay, bache? Right, so when it comes to the cell, plant cell, right, they have the cell wall. So cell wall, it will start in the center, it grows outward. Okay, bache, so cell plate will form here. So at the time of cytoplasmic division, organelles like mitochondria and plastid, they will get distributed between the two daughter cells, and sometimes when karyokinesis is not followed by cytokinesis, you know that we will get cenocytic condition. Okay, cenocytic condition, jaise endosperm in the case of coconut. Baki significance of mitosis mein, you know that it is the equational division, usually restricted to diploid cell, but you know that even haploid cells also show this, right? So it results in the production of diploid daughter cells. The growth of multicellular organism is possible due to this. It helps in distributing the ratio between the nucleus and the cytoplasm, right? It therefore becomes essential for the cell to divide. So here dekho, cell growth will do what? Cell growth will disturb the ratio in between nucleus and cytoplasm. So division will ensure that it is proper. We have already covered it. So the baki cells of upper layer of epidermis, lining of gut, blood cells, they are constantly replaced because of the division, and that's it, bache. That's it. So now before starting meiosis, I'll ask you a few questions. Ye dekho, jaise let's say this nucleus is in prophase. So tell me the amount of DNA here. Tell me the amount of DNA here. Tell me the amount of DNA here, if it is in prophase. And let's consider ki it is having 4 nucleus. Ye dekho bache, sorry, 4 chromosome. Bache, it is going to be like this, no. So it is double, it is 4C. Now let's say if it is a telophase. Now you know that this is the situation. Yes, this is the situation. Am I right? This is the situation. So if this is the situation, chromosome number is same. Here also, it is 4. In the daughter cells also, it is going to be 4. But look at the amount of DNA. Please focus here. Done? Done? ठीक है? Okay, चलो next. बच्चे, next is the meiosis. Very important division. Reductional division it is. If in this division, if you understand what prophase 1 is, then everything will be very easy for you. ठीक है? So production of offsprings by sexual reproduction, it needs the fusion of gametes. And if these gametes, they are not going to have haploid cells, how do we maintain the diploid state in the zygote? Right? We need that. We need that for diploid organisms. And overall, you know that zygote is going to be diploid. Right? So gametes are formed from specialized diploid cell. So they're specialized kind of cell division that reduces the chromosome number by half is your meiosis. So it will ensure the production of haploid phase in life cycle of sexually reproducing organisms. Just even in our case, normally our body is diploid, but our gametes are haploid. And how is it possible? Because of meiosis. Because of meiosis. And ultimately after meiosis, bache, fertilization is there that will restore that diploid phase. Right? That will restore that diploid phase. Is that clear? Is that clear? So meiosis is important during gametogenesis in plants and animals. We will see this, right, bache? So this is what we have in meiosis. So when you talk about the meiosis, remember one thing. It will take place in 2 rounds, right? So it means 2 times nuclear division, that is your karyokinesis, 2 times cytoplasmic division. Very important it is. 2 times nuclear division, 2 times cytoplasmic division, but DNA replication occurs once. DNA replication, it occurs once. Is that clear? Is that clear? Just say, see, this is a diploid cell. It is undergoing meiosis 1. So if it is undergoing meiosis 1, definitely karyokinesis will occur. Definitely cytokinesis will occur. So in meiosis 1 again, the trick is same. That is PMAT. The trick is same. That is PMAT. The trick is same. And what is that trick? The trick is PMAT. There will be prophase 1. There will be metaphase 1. There will be anaphase 1. There will be telophase 1, and obviously after it there will be the I'm writing it here obviously for firstly karyokinesis and then cytokinesis will occur indeed, and you will get 2 haploid daughter cells. 2 haploid daughter cells. Now that 2 haploid daughter cells, they will undergo meiosis 2. Here also the story is same in karyokinesis. That is, here also you will see the trick is same. It is prophase 2. It is metaphase 2. It is anaphase 2. It is telophase 2. Telophase 2. And finally, we will get 4 haploid daughter cells. Now what's the point? Dekho, initially here, because it is taking place after interphase, here the DNA amount is going to be 4C. DNA amount will be 4C. After meiosis 1 in these 2 haploid cell, the DNA amount is 2C and 2C. But we do not want that much of DNA. We have to make it C. Let's say If here it is 100, in these two daughter cells it will be 50-50. Here finally it will be 25, 25, 25 and 25. Okay, okay, okay. And normally why 25, 25? I'll tell you. Let's say this is a sperm, that's ovum. It should be spermatozoa and ovum, haploid, haploid. They will fuse, they will form diploid zygote. They will form diploid zygote. So when it is a diploid cell, obviously here the DNA amount should be 2C. It should not be 4C. We are not accepting that. We normally in a cell we want this as the DNA amount. Okay? So sperm, haploid, haploid. So here the DNA amount should be C and C, and that will make it 2C. Right? So again observe it. Let's say if 100 gram DNA is there, after meiosis 1, 2 daughter cell will have 50-50, but we want 25-25 because ultimately 50, matlab 2C, it is the initial amount. It is the initial amount, and that is what we need. Are you getting my point? That is what we need. So this is the meiosis, right? So 2 times nuclear division, 2 times cytoplasmic division, but DNA replication will occur once. Right? So again, I'm telling you how will it proceed. Rest everything is same. Interphase it is, there will be G1, there will be S, there will be G2. After G2, it will enter in meiosis 1. And as I said, when it is meiosis 1, students, prophase 1, metaphase 1, anaphase 1, telophase 1. So after this telophase and before starting of this meiosis 2, yeah, you can say that prophase 2, so this gap here is known as inter, means in between, kinesis means division. In between division there is a gap, right? This is how this is how it is. So it will start from the same thing, interphase G1 S, where DNA content will get doubled, but number of chromosome will remain same. But number of chromosome will remain same, right students? Right students? So here there will be prophase 1, metaphase 1, anaphase 1, telophase 1. After telophase 1, it's meiosis 2. Prophase 2, metaphase 2, anaphase 2 and telophase 2. So in between telophase 1 and prophase 2, there is a very short gap, that is interkinesis. Right? There is interkinesis, that gap. But it is not like interphase. Right? No DNA replication is taking place. DNA replication will occur only in the S phase initially. It will occur only in the S phase initially. So up to this part all clear? Yes, tell me. Up to this part all clear? Yes, please. Tell me. Up to this part all clear? Anji, any doubt? Yes, do you have any doubt here? Or is it clear? Yes? Done? Is it clear or not? Okay? Okay, right? So let's start. Okay? So now when we talk about the prophase one, it's just like homework, right? Then we will start the actual topic. In prophase 1, there are 5 substages. Do you know that? 5 substages. And there is a trick for that. That is L, Z, P, D, D. Repeat it. L, Z, P, D, D. Right? L, Z, P, D, D. So it can also be like this. Little Zara, please do not dance. Little Zara, please do not dance. Okay? So L, Z, P, D, D. So it is leptotene, it is zygotene, it is pachytene, it is diplotene, and it is dikinesis. And let me tell you, it is a favorite question of your examiner. For sure, you will get a question from this part. So it is LZPDD. It is LZPDD. So it is leptotene, it is zygotene, it is pachytene, it is diplotene, it is dikinesis. It is dikinesis. Okay? Leptotene, zygotene, pachytene, diplotene, dikinesis. Leptotene, zygotene, pachytene, diplotene, dikinesis. So it is leptonema. Sometimes also this is also zygonema, pachinema, diplonema. These are also the words that we can use here. Okay? We can use here. Okay? So leptotene, zygotene, pachytene, diplotene, dikinesis. Right? Right? Dikinesis. Clear? Sure? Are you sure? Are you sure? Then we'll tell you about the checkpoints. Okay, so please note down. Hope it is done. चलिए, let's go. Okay, चलिए, very good. So ये देखो, that's what we have. Meiosis 1, Meiosis 2, and the gap in between, जैसे ये देखो, this gap in between telophase and prophase 1, that will be your interkinesis. No DNA duplication is taking place here. This is your interkinesis. This is your interkinesis. So now we have to start the prophase 1. Okay, prophase 1. So obviously it is the longest phase. It is the longest phase. Okay, what's that? It is the longest phase. Ultimately, condensation of chromosome will be there. Nuclear membrane will disintegrate. But here we will also see some other things. देखो, ultimately what are we doing? We have a diploid cell, right? And after meiosis 1, we are forming 2 haploid cells. Okay? So I'll give you the example. For an example, let's say this cell is having 4 chromosomes. This cell is having 4 chromosomes, right? So after meiosis 1, we have 2 daughter cells. After meiosis 1, we have 2 daughter cells, and that daughter cells are having n, matlab n number of chromosomes. So can I say that 2 chromosomes? Can I say that 2 chromosomes? 2 chromosomes. And after meiosis 2, again, you know what? Meiosis 2 is considered as equivalent to mitosis. Do you know meiosis 2 is considered as equivalent to mitosis? Why? Because here also the daughter cells are having same number of chromosomes just like parent cell. But how is it possible? That's how it is possible. Ultimately, it is to divide the DNA content. That is the case here, right? You firstly observe it and then I'll explain. That is the case here, right? Done? Done? Yes? Is it clear? So that's the case. Dekho, so we have to separate the chromosomes. Ultimately, here in the prophase, matlab one by one we will discuss it, but let me tell you, here we will see some new things that were not there in the mitosis. Just say here we will see pairing of homologous chromosome. We have to pair, matlab, let's say in humans we have 46 chromosomes. Right, we have 46 chromosomes, right? It means 23 pairs are there. 23 pairs are there, right? So in our daughter cells, matlab in the gametes, ultimately what do we want? We want 23 chromosomes, no? So we have to separate these pairs. So firstly in the meiosis, we will pair the chromosomes, right? Homologous chromosomes of course, and then we have to separate them. We have to separate them, right? In the mitosis, there was nothing like that. 4 chromosomes are there, all of them were aligned, right? You know that sister chromatids were getting separated and they will start behaving like the chromosome and the daughter cell will have the same number of chromosomes. But here you have to reduce the chromosome number to half. Here you have to reduce the chromosome number to half. So here, there are some extra things that we have to discuss, and that is why this meiosis, because meiosis is making the gamete. Right, and that gametes are required for sexual reproduction, and you know that sexual reproduction is advanced, right? It is better than the, right? This sexual reproduction is better than the asexual reproduction. Why? Because we use a word, I hope you know that, variations. That due to sexual reproduction, variations are there, changes are there, and that variations are possible due to 2 reasons, bache. One is mutation, sudden changes in our DNA. Another is recombination. We call it as crossing over. So during meiosis, this recombination, this crossing over will occur. So right now I'm giving you the overview. Then one by one we will discuss it. So when we will start this particular topic, that is prophase 1, we will see what is happening in different substages. We will see that chromosome condensation will start. Then we will see that the pairing of chromosomes will be there. It's a very important thing. It's a very important thing here. We have to pair the chromosome, right? We have to pair the chromosome. Then we will see the recombination. Just I'll give you the example. We are not ditto like our mother or like our father. Just if you talk about me, I have the character of my mother as well as of my father. Just say my eyes are just like my father, my nose is just like my father, right? My complexion is more like my mother. Are you getting my point? Are you getting my hair are more like my mother? Okay, but my height is like my father. So basically, I am the combination of my mother and father. I have the character of my mother as well as of father. It can vary. These things can vary. Let's say I'm more like my father, right? His characters were dominating. I'm expressing them, right? I'm less like my mother. That can be the possibility, but I am not saying that I'm ditto like my father. Right, that ditto kind of thing can only happen when there is the asexual reproduction or linkage is there, and that too complete linkage, where chromosomes are staying as such in the parent cell and the daughter cell or the gametes, right? It is not at all changing. That is very rare. That complete linkage thing is very rare, right, bache? Right. So what am I trying to say? I'm trying to say that there are some new things that we have to understand. Metaphase is going to be same. Anaphase is going to be same. Telophase is going to be same. Few changes will be there, but ultimately it is same. Metaphase mein we have to align the chromosome at the equator, right? It's just that ki metaphase 1 mein you will have 2 plates. I'll tell you. In anaphase we have to separate it again. The chromosome pair will get separated. That type of things are there. So initially, when we start this process, we'll see that we have to pair the chromosome. And as I said, we are not ditto like our mother or father, right? We are the combination of both. We are the combination of the both. So we will see the crossing over will occur, right? This is some part of maternal chromosome will exchange its segment with the paternal chromosome. That type of things will be there. Are you getting my point? That type of things will be there. Are you getting my point? Are you getting my finally after that we will see, right, that chiasmata formation, the terminalization of chiasmata, and finally the metaphase. So ultimately in the meiosis one, our target is pairing of homologous chromosome, crossing over, then terminalize that chiasmata formation and terminalization of chiasmata. And by the end of the meiosis 1, we want our daughter cells to have half the number of chromosomes than the parent cell, right? And meiosis 2 is just like mitosis. You are going to grab it like this. Okay? So that's what we have to read. So I am giving you a break, but this time this break is for 15 minutes only, because after 15 minutes you have to give me your 30 minutes and the topic will be over. Trust me, it is the prophase 1 actually that you need to understand, right, where you have to be very focused. If your concepts are clear, metaphase easy, anaphase easy, right, and the comparison with the meiosis 2 very easy. So right now I'm giving you a break. It's 10:50. So break time is till 11:10, right? So 20 minutes ka break. So from 11:10 to 12 maximum, we'll finish this topic simple. We will finish this topic simple. Okay? We'll finish it. Hello. Hello, hello. Now am I audible? Now mic testing 1 2 3. Now am I audible? Hello. Hello. Okay. So it is the complicated and the longest in the duration. Okay. Longest in the duration. Is that clear? Is that clear? So obviously there are sub stages that we need to discuss. Okay. So here you can say, matlab after prophase 1, cell size will get doubled. It will be doubled, right? The cell size, it will get doubled, and volume of nucleus will be about 2 to 3 times, about 2 to 3 times. That will be the changes. Okay, so let's start with the leptonema first. Okay, so leptonema or leptotene it is, and obviously it is the first sub-stage, bache. So it will start with the condensation of the chromatid. How is it going to start? It start with the condensation of the chromatid. So obviously chromosomes, ultimately what will happen to the chromosomes here? Chromosomes, they appear, yes, chromosomes, they appear thin, right? They appear thin and obviously elongated, uncoiled, right? And but they'll be, just see, if they are thread like, at points now they'll be having some beads, they'll appear beaded also. Here na chromosomes, they appear beaded means just like they are having some beads, just this structure, it is chromomere, it is chromomere, it is chromomere, it is beaded. What's that? It is beaded appearance is there. It is thin, it is elongated, it is uncoiled, and it is having some beads like structure, and they represents gene, right? It represents these chromomeres, it represents gene. Are you getting my point? It represents gene. Are you getting my point? Are you getting my point? Right? So if you think that by this stage the chromatids will become clear, no. Chromatids are not visible yet. They are not visible here. Okay? And do you know this leptotene stage? This leptotene stage is also known as the bouquet stage, especially in some insects. It is also known as the bouquet stage, right? Due to the characteristic appearance of the chromosome. I'll tell you what is that. Appearance of chromosomes. Are you getting my point? Right, so bouquet stage. So it is due to the characteristic appearance of chromosome. Actually, what is happening in this particular stage? Just imagine the chromosomes from both their ends, right? They will attach to this nuclear envelope or to the matrix here, right? You can say that these chromosomes, they'll get attached here, right? They will attach to this nuclear lamina, your nuclear matrix, with the help of their ends, right? Basically, you can say that both the ends of the chromosome, by this time they are not even clear, they are not even discrete, but that ends of the chromosome, they will attach to a structure, that is the attachment plate, right? That structure is the attachment plate, and with the help of that attachment plates, they are attached to this nuclear lamina. Okay, they are attached to this nuclear lamina. Are you getting my point? They are attached to this nuclear lamina. Is that clear? Is that clear? Yes. So chromosomes, they will attach to the nuclear lamina by its end through attachment plate, bachche, right? Is that clear? And do I need to explain that? Yes. Do I need to explain the same thing again? That obviously centrioles are present here, they are duplicated, they will move to the opposite pole. Matlab that thing will also occur, right? Because it is also the prophase. So the compaction of chromosome, that will compaction of that chromatin, ya fir you can say condensation of chromatin will be there. But here in this leptodene is also known as the bouquet stage due to the characteristic shape of the, right, due to the characteristic shape of the chromosome. So what is happening? The chromosome by their ends, they are attached to the attachment plate and then to the nuclear envelope. Is that clear? Then to the nuclear envelope. Is that clear? Is that clear? Hanji, is that clear? Sure? Sure. And do remind me that I need to teach you the checkpoints. Okay? So leptodene clear? Leptodene clear? Right? So a group of chromosome in the here in the nucleus, they'll appear like a bouquet. So it is the bouquet stage. So this is the first stage, and obviously compaction will occur. Compaction occurs throughout the prophase, right? It's not like that here in this particular substage the complete compaction will occur. Obviously not. The compaction will occur throughout the prophase, right? The compaction will occur throughout the prophase. Is that clear? Is that clear? Done? Okay, chaliye. So this is the leptodene. Next is the zygotine. Next is the zygotine. See, already you know that I told you already that we have to pair the homologous chromosome, then the crossing over will occur. So that pairing of homologous chromosome is known as synapsis or syndesis. Synapsis or syndesis. So that will take place in the zygotine, right? That will take place in the zygotine. Are you getting my point? So when you talk about the zygotine, what is happening there? It is the synapsis or the syndesis. So synapsis means pairing of homologous chromosome, right? It means pairing of homologous chromosome, right? So which chromosomes are homologous chromosomes having same centromere position, same size, same shape and same gene position, right? The genes are present at the same locus there. The pairing of homologous chromosome will be there in this particular substage. So just say, let's say this is your paternal chromosome, right, from father's side. This is your maternal chromosome from mother side. They will be paired. They'll come closer. They will be paired. Right, there are some other proteins also that we discuss, cohesin, condensin. They will also help in the compaction and all, but obviously not the part of your syllabus, so I'm not touching this. Right, so pairing of homologous chromosome will be there. Now see, this greenish, they are attached to the same centromere. They are attached to the same centromere. They are sister chromatids. Am I right? They are sister chromatids. These two pinky, they are also sister chromatids. They are also sister chromatids. Are you getting my point? They are also sister chromatids. But here, because it is a pair of chromosome. One chromosome is from mother's side, another chromosome is from father's side. Right? I have already explained what homologous chromosomes are. Homologous chromosome, it's a pair of chromosome in which one chromosome is given by the mother, one chromosome is given by the father. These chromosomes are having same centromere position, same shape, same size, and the gene position is also there. Right, the same gene position is also there. Is that clear? Is that clear? So look at this, right? Just look at it. Okay, so these two, they are the non-sister chromatids. What are they? They are the non-sister chromatids of homologous pair of chromosome. These two are non-sister because they are not attached to the same centromere. Understood? So this is zygotine, right? The pairing of, yes, the pairing of homologous chromosome, right? The pairing of homologous chromosome is there, right, bache? Right? Yes or no? right? So this is synthesis, this is synapses, and we can also call it as zipping. We can also call it as zipping. Now you tell me, if we have 46 chromosomes in totality, then in the zygotine, right, how many pairs are we going to see? How many pairs are we going to see? Because there is a pairing, right? There is a pairing of homologous chromosome. So how many pairs are we going to see here? How many pairs are we going to see here? How many pairs are we going to see here? Yes? Hello? Obviously 23, half the number of chromosome, 23 pairs will be there, right? So how these pairs they'll be stabilized? Just imagine, this is your one pair of chromosome, this is your another pair of chromosome, right? So we have to do their pairing. So it's not like that you will tell them, "Arey, stay, right, stay closer, yeah, stay together." Obviously not. We have to stabilize the structure as well, na? So in between, bache, basically how that pairing will occur, right? You will see a complex that will form in between. So question can also come from this complex, right? And what is the name of that complex? It is synaptonemal complex. It is synaptonemal complex. What's the name of complex? It is synaptonemal complex. What is the name of complex? It is synaptonemal complex. Are you getting my point? Yes, are you getting my point? What is the name of the complex? It is the synaptonemal complex, and that is the one that is helping in the pairing. Just say you can imagine it like this, कि this is another chromosome. So in between these two, this is the synaptonemal complex. ठीक है बच्चे, this is the synaptonemal complex, right बच्चे, right बच्चे, right? So it is a ribonucleoprotein complex. Okay, it is a ribonucleoprotein complex. ठीक है, ribonucleoprotein complex. So this protein complex is having RNA, it is having proteins, and on its both sides, right? And here on its both sides, obviously the lateral element is basically the DNA is also there. So this synaptonemal complex, which is a ribonucleoprotein complex, it is going to stabilize it. Is that clear? It is going to stabilize it, right bache? So one central RNA, protein are present, right? So lateral element is having DNA, RNA and protein. So it is in between the sister chromatids, right? It is present between the sister chromatids here. Okay? Okay, so it is going to stabilize it. Any doubt? Right? Any doubt? Yes? Yes? Any doubt? So done. So pairing of homologous chromosome will be there. And let me tell you, bache, right? Let me tell you, this pairing continues till metaphase 1. Matlab these chromosomes, they'll be paired till metaphase 1. Till metaphase 1. Right. So now see, two pairs. You have a pair of chromosome. You have two chromosomes here held together with the help of synaptonemal complex. So synaptonemal complex is formed in between the homologous chromosomes, or you can say that in between these sister chromatids having RNA, protein and the DNA also. Right. So bache, it's a pair, na? So can I call it as a bivalent? Can I call it as a bivalent? Bivalent. Can I call it as a bivalent? Yes, it is a bivalent. Yes, definitely I can call it as a bivalent or tetrad. Why tetrad? Because it is a pair, right? Two chromosomes are there. I'm using the word bivalent here. It's a tetrad. Tetra mane 4. Tetra mane 4. So here if you will see, two sister chromatids, two sister chromatids. So total 4 arms are there na, ya 4 sister chromatids are there na. 4 sister chromatids are there na. So bivalent and tetrad is same. Bivalent and tetrad is same. And this bivalent or tetrad is clearly visible in the next substage, that is packaging. So leptotin mein normal condensation will start, right? Plus bouquet stage will also be there. Matlab we'll see the bouquet stage also because of the characteristic appearance of the chromosome. The ends of the chromosome will attach to an attachment plate. With the help of that, it is attached to the nuclear envelope. Then zygote, right? Right, definitely you will get question from this part. Synapses, the pairing will be there. And for that pairing, because we have to stabilize this structure, so synaptonemal complex will form. Synaptonemal complex will form, right? Synaptonemal complex will form. So you should know what a homologous chromosome is, what are the sister chromatids, what are the non-sister chromatids. It should be crystal clear to you all. It should be crystal clear to you all. Is that clear? Chaliye. Now what's the next thing? Next thing is packeting. In comparison to these two substages, packeting is comparatively longer, right? In comparison to these two substages, right? The first two stages of prophase, they are short, right? Compared to this stage. Compared to this stage. So first two sub-stages of prophase 1 are short compared to this stage. This is the packeting. And what is going to happen here? Very important thing responsible for variation. It is your recombination, guys, or also known as crossing over, or also known as crossing over, right? Crossing over. Re means again, to combine again, right, bache? Right, recombination, new combinations you will see here, and the enzyme used here is recombinase. So what is going to happen, bache? There will be an exchange of segment. I'll demonstrate it as well. There will be an exchange of segments. There will be an exchange of segments in between. This point is very important. There is a previous year question from this part. There will be an exchange of segment in between non-sister chromatids. The word is non-sister chromatids of homologous pair of chromosome. This is important, bache. Please try to understand. Whenever we talk about the crossing over, it will always be there in between the, see, this is the homologous pair of chromosome. So crossing over will be there in between the homologous pair of chromosome and in between these non-sister chromatids. Sister chromatids are not going to exchange their segments. Non-sister chromatids will be. Let's say it is your maternal chromosome, this one is your paternal chromosome, right? So they will exchange their segments. So obviously we don't discuss that in detail, but at least you should have an idea of it. Here we also see that let's say this chromatid is exchanging with the segment with this one, double crossovers, single crossovers, that type of terminology is there, but obviously not the part of your syllabus, and if I will start, you people will start crying. So ye dekho, this non-sister chromatid and these two non-sister chromatids, they will exchange their segment. Which enzyme is playing the role with their recombinase, right? Recombinase, right? Recombinase. It will cut and it will then join these segments. So now next is what? Next is what? Just imagine. See this blue one will get this color. That's how the segments will get exchanged. Suppose this is my maternal. Okay, so see, this is my maternal chromosome, right? A fragment of it, a segment of it will get exchanged with my paternal chromosome. So now this chromosome is having these alleles, right? Right, bache? That's how it will get exchanged. This is the crossing over. Now, if you will observe here, right? This is the site. ये देखो, let's say crossing over will occur, recombination it is. It is the base of variation. We say that because of sexual reproduction, variations are there. And why? Because of recombination. So these are this is the homologous pair of chromosome. These two are the non-sister chromatids. These two are the non-sister chromatids. Right now they will exchange their segment. Let's say if at this place, if at this point the recombination has to occur. So we call this point, so we will see a recombination nodule here, right? Nodule means some kind of there's a node, some like some condensed kind of structure. They are ganthe nodes, some nodes are there. Just say you can compare it with the stem, nodes are there that aggregated such structure, right? Something like that. So what am I saying? That see, this is the maternal chromosome, this is the paternal chromosome. Let's say this is the site at which the crossing over is destined to occur. So it will be having the recombination nodule. So recombination nodule represents the site at which crossing over occurs. Recombination nodule, stella, it represents the site at which crossing over occur. Now, the segments, they will get exchanged. Recombinase enzyme will play the role. Now, let's say this is the site. Now, the recombination is done. It's completed. Still, these two segments, they'll remain attached to each other. They will not separate. So, we will call it as chiasmata. 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 What is the word that we are going to use here? Chiasmata. What is the word that we are going to use here? It is chiasmata. Are you getting my point? Chiasmata. Let's say this is another pair. So it is exchanging the segment. Okay. In my diagram, it is not appearing like X. Sorry. It is not looking like a X, but it is, one second, something like that. Okay? Right? So now they are exchanging their segment. So the site at which they have exchanged their segment, right? That X-shaped structure will appear here. X-shaped structure will appear here. I know it is not looking like X shape, but still X-shaped structure is appearing there. We are calling it as chiasmata, right? So recombination nodule is the area which is representing the Hanji. Here at this point the recombination will occur. After that recombination, after the exchange of segment, after that recombination, after the exchange of segment, right? After the recombination, after the exchange of segment, what are we going to see here? Right? We will see the X-shaped structure. So it represents the site. Matlab, still, right? Still, these two chromosomes, right? Just let's say here they exchange the segment. So we will see that there will be a X-shaped structure. We are calling it as chiasmata. So is that clear? Is that clear? So always remember, there is the appearance of recombination nodule that represents the site at which the crossing over occurs, and crossing over will be there in the non-sister chromatids of homologous pair of chromosome. Just this is one homologous pair of chromosome, this is another. It's not like that this chromosome will exchange its segments here. No. This is one homologous pair of chromosome, this is another homologous pair of chromosome, right? So they are non-sister chromatids. They say this one can also exchange segment with this one can also exchange segment with this one. Matlab, single crossover, double crossover can be there. Just this one with this one, this one with this one. Okay? That is the possibility. That is the possibility, right? That is the possibility. But it's not like that this chromosome will exchange its segment with some other homologous pair. No. So again I am repeating, non-sister chromatids of homologous pair of chromosome, they exchange their segment, and before crossing over a recombination nodule will appear. That is marking the site at which the crossing over will occur. Okay? Is that clear? So crossing over, enzyme mediated process, recombinase is the enzyme that will be used. Okay? Right? So even after the recombination, chromosome will still linked at the site where crossing over has been done, and that is forming X-shaped structure, chiasmata. Any doubt? Any doubt? Any doubt? Yes. So next substage is diplotin. Diplotin means dissolution of, yes, diplotin means dissolution of synaptonemal complex. And again I'm telling you, this is a favorite question of your examiner. Okay, ji? So diplotin means dissolution of synaptonemal complex. What will be there? There will be the dissolution of synaptonemal complex, right? So basically the synaptonemal complex in between these two will dissolve, right? The synaptonemal complex in between these two will, okay, will dissolve, okay, it will dissolve, okay, बच्चे. But I told you that pairing will be continued, pairing will be continued till metaphase 1, right? The chromosomes will be in the paired stage till metaphase 1. The chromosome will be in that paired stage till metaphase 1. So no doubt the synaptonemal complex in between it will dissolve, but still this pair of chromosome, this bivalent, actually in Pacatina now that bivalent is very clearly visible. That bivalent is very clearly visible. It is tetrad. It is that sister chromatids are very clearly visible in Pacatina. Okay, so this bivalent will still remain attached to each other, right? How? With the help of that X-shaped chiasmata. With the help of that X-shaped chiasmata, right? With the help of that X-shaped chiasmata, it will stay. Still, they'll join to each other. Okay. Now the next is your dikinesis. And dikinesis is characterized by terminalization of chiasmata. देखो, जैसे crossing over. Let's say it occur at this point या फिर this point. It occur at this point or this point. So in the dikinesis, this chiasmata, this is the X-shaped structure which is showing that this is the site where crossing over has been done. So there will be the terminalization. Terminals means ends. Means ends. What is the meaning of terminalization? Terminalization of chiasmata. Terminals mean ends. So what is the point here? See, let's say if this chiasmata is present at this particular position, so in the dikinesis, terminalization of chiasmata, that X-shaped structure will come to the ends. It will come to the ends. Okay? Just let's say these two chromosomes, non-sister chromatids, they are exchanging their segment at this point. Okay? Before exchange we will say, let's say if this is the site at which crossing over occur, so before exchange there is the recombination nodule that will appear here. After exchange, because they are exchanging their segment, we will see that an X-shaped area, X-shaped structure, right, called chiasmata will form. Right now, let's say it is forming at this point. So in diplotin, the synaptonemal complex in between will dissolve, but still chromosome will stay attached to each other with the help of this chiasmata. Now in the dikinesis, there is terminalization of chiasmata. If that chiasmata was here, it will come to the ends. And why is it still here? Because we know that we have to, right, keep the chromosome in the paired form. We have to keep it, right, till metaphase 1. They'll stay like this in the paired form. Understood? Okay? Chalo, NCERT Didi. Still, if there is any doubt, please feel free to ask. Now let's talk about NCERT and then we will talk about the metaphase 1. Trust me, if prophase 1 is clear, I'll explain what is going to happen in the metaphase 1, then everything is easy. Then I just need your next 15 minutes and the topic will be over. ये देखो, जैसे for an example, if a cell, let's say it is having 8 chromosomes. Let's say it is having how many chromosomes? 8 chromosomes, right? 2n is equals to 8. So after this prophase, after this, one minute. So after prophase, because the pairing of chromosome is there, no? So you are going to see 4 pairs, am I right? 4 bivalent या tetrad, yeah, these diets, that's what you are going to see. Total 8 chromosomes are there. After this zygotine, that's what we are going to see, the pairing. Means even by the end, By the end of prophase, chromosomes, they'll become compact and they'll be in this paired state only, na? Even in the dikinesis, there is a terminalization of the chiasmata. Chiasmata will still be there at the ends. This is the case. This is the scenario. And you know that because it is the prophase. So ultimately, even nuclear membrane will also disintegrate. All the chromosomes, these pairs, they will be in the cytoplasm. Because what is the feature of, Sujay, what is the feature of chiasmata? What is the feature of your metaphase? In the metaphase, all the chromosomes are going to align themselves at the equator, right? At that metaphasic plate. So here we will see it is not the individual chromosome. We have the pairs. So even these pairs are going to arrange itself, right? Because that is going to happen in the metaphase for sure. Metaphase may ultimately chromosomes, they are going to arrange itself at that. That particular chromosome is going to arrange itself at that equator. And because here we have the pair of chromosome, so in metaphase 1 of in the metaphase 1 of meiosis 1, you will see that pair will come, right? Right? So let's complete this part first. So prophase 1, it is of the first meiotic division, is typically longer, more complex when we compare it with the prophase of mitosis. Okay? Then let's talk about zygotine. So what is happening in zygotine? The chromosome become gradually visible under the light microscope. Important point. It's a previous year question. Chromosome, we will see it under light microscope. The compaction of chromosome continues throughout the leptotene. Then zygotine will come, and in the zygotine, synapses will occur. So synapses, synthesis, zipping, it's all same. Synapses, synthesis, zipping, it's all same. Means pairing of homologous chromosome. So these paired chromosomes are homologous chromosome. Electron micrographs of this stage, under that electron microscopes and all, you can see that this pairing is due to the formation of synaptonemal complex. So this synaptonemal complex form in between the synapsed homologous chromosome, right? So it is the bivalent or the tetrad. Doubt. Bivalent or the tetrad. Doubt. 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 Yes. Next is packetine. This packetine is comparatively of long duration. Here, bache, 4 chromatids of each bivalent. Right, tetrad it is, no, it will become distinct and very clearly they'll appear as tetrad. Now in that pair, the tetrads are very clearly visible. That 4, that chromatids are clearly visible, right, bache? So here they are saying that there will be the appearance of recombination nodules. There will be the appearance of recombination nodule. So recombination nodule is the site at which the crossing over occur between 2 non-sister chromatids of homologous chromosomes. Is that clear? Is that clear? So crossing over, what exactly is the crossing over? It is an enzyme mediated process. It is the exchange of genetic material between 2 homologous chromosomes. Okay? Between 2 homologous chromosomes. So crossing over is an enzyme mediated process. The recombinase enzyme will be used here. So recombination between homologous chromosome is completed by the end of packetine, leaving the chromosome linked at the sites of crossing over, and that is basically chiasmata. Any doubt, Sujay? Yes, bache, Vrinda, Shreya? Any doubt? Okay. Next, the beginning of diplotin is recognized by dissolution of the synaptonemal complex. So if dissolution of synaptonemal complex is there, that bivalent will try to separate. They will try to separate, but still they'll stay connected. Why? Why? So overall, देखो, जैसे synaptonemal complex was there, they were attached to each other, but now there is the dissolution of synaptonemal complex, but still, so chromosome will try to get separated, but still they'll stay connected at the site of crossing over, that because of chiasmata. Right, just say they were like this due to synaptonemal complex. Just imagine now, because synaptonemal complex is dissolved, so they'll try to get separated, but still they'll stay connected to each other at the points where crossing over has been done. Okay, because it is the X-shaped chiasmata. Okay, it is the X-shaped chiasmata. Right. So now in the oocytes of some vertebrates, diplotin can even last for years. जैसे let's consider the example of even humans. But in our case, when we talk about oogenesis, right, it will start during embryonic stages. Right, it will even start during embryonic stages, oogenesis. So basically oogonia, right, that cells that are going to form the female gamete, oogonia, that oogonia will grow, they'll form primary oocyte, and that primary oocyte, even in the fetal stages, even during the fetal stages, it will start the meiosis. But again, there will be a block or a halt at the diplotin stage, and that is known as the dictate arrest. So after birth, from birth to puberty, many follicles in our ovaries, they'll degenerate. At the time of puberty, these primary oocytes, under the hormonal influence, they'll start growing. Their meiosis 1 will continue, and then metaphase 2 arrest will be there. So that's why they are saying in some oocytes, right, of some vertebrate, diplotin can even last for months or even for years. And we also take the example of amphibians there, right? We also take the example of amphibians there, right, bache? Right. So final stage of meiotic process, phase 1 is diakinesis, where terminalization of chiasmata is there. So chromosomes are fully condensed by this stage, bache. Overall, obviously, centrioles, they are at opposite pole. Meiotic spindle is ready, right, bache. So it is assembled to prepare the homologous chromosome for separation. So by the end of diakinesis, again, just like your mitosis, nucleolus will disappear, nuclear envelope will break. So diakinesis represents transition to metaphase. It's important. It represents transition to metaphase. It's important. Okay? This is important. Okay. Now next is the metaphase 1. But rest everything is same. ये देखो. I told you, just take the example of a cell having how many chromosomes? Having 8 chromosomes, right? Having how many chromosomes? Having 8 chromosomes, right, bache? So obviously, because they are paired, right? So this pair will arrange itself here at the equator, right? This pair is going to arrange itself at the equator. So whenever they'll give you the picture, right, just look at this. You can very clearly say, this is dekho. If it will be this is the metaphase 1. What's that? It is the metaphase 1 of prophase 1. Sorry, of meiosis 1, right? Then there is the metaphase of mitosis. Let's say we are taking same example that this cell is having total 8 chromosomes. So when it is the meiosis, right? When it is the meiosis 1, right? Because we have to reduce the chromosome number to half. So that is how that 8 chromosomes will get arranged. But when it is your mitosis, right? I'm using only one color here. Okay? So that is how your chromosomes will that's how they are going to arrange. Are you getting my point? So in the metaphase 1, there will be 2 metaphasic plates because bivalent is arranged here. Because bivalents, they'll come to this side. And in the metaphase of mitosis for the same cell, that will be the scenario, right? Your eight chromosomes, they will align. Let's say your 8 chromosomes, they will align. Is that clear? Right? Is that clear? So just look at the diagram. Just look at the diagram and you'll get to know the answer. Right? So now imagine. In the anaphase of mitosis, what will happen? Right? When you talk about the anaphase, just look at it. You know that ultimately in the anaphase, the separation will be there. They need to move to the opposite pole. So when it is the anaphase of mitosis, because again, it is also a repeated question. Right? It is also a repeated question. So when it is the anaphase of mitosis, there is the splitting. There is the splitting of centromere. There is the splitting of centromere. And there will be separation of sister chromatids. There will be separation of sister chromatids. Am I right? Am I right? ये देखो, ये देखो, right? There is the splitting of what? There is the splitting of centromere and there is the separation of sister chromatids. There is the separation of sister chromatids. Yes or no? But now let's talk about the anaphase 1 of meiosis 1. Let's talk about the anaphase 1 of meiosis 1. Right? Let's talk about the anaphase 1 of meiosis 1. Done? Okay, so when it is the anaphase 1, when it is the anaphase 1 of, right, when it is the anaphase 1, see, now obviously we have to separate the bivalent. So there will be the separation, please try to understand, there will be the separation of bivalent, right? So bivalent chromosomes will separate. So there is no splitting. There is no splitting of centromere. There is no separation of sister chromatid. No splitting of centromere. No separation of sister chromatid will be there. Just let's say if this is the pair, obviously they are after crossing over they will form. Right? After crossing over they are going to form. They have exchanged their segments. Right? So it is like this only. It is like this only. It is like this only. ठीक है बच्चे? ठीक है. Okay. So in the anaphase 1, there will be the separation of bivalent. Now you imagine it. In the telophase. No doubt, right? In the telophase, no doubt, this cell will contain 4 chromosomes. Right? You know, na telophase mein it will reach to the opposite pole. It is just reverse of. Right, it is the reverse of prophase. So don't you think this will be the situation? This is going to be the situation. Right, that is why bache we say that after telophase, right, daughter cells are haploid and haploid. Right, but look at the DNA content. Initially it was 4C, but here it will be 2C, We have to keep it C. Right, we have to keep it C. Done? Done बच्चे? Done? Is it clear? Is it clear? Is it clear? Yes or no? Right? So that will be the case. Now after this telophase, see, let's say, obviously after telophase again cytokinesis will occur, right? After telophase again cytokinesis will occur. After telophase again cytokinesis will occur. Obviously we will get two daughter cells, right, having 4 chromosomes. Okay, having 4 chromosomes. Okay, now bache, see, now there will be interkinesis. A kind of gap will be there. Matlab in this gap the DNA replication will not occur, and after this there will be prophase 2. So we compare, right, we compare meiosis 2, right, we compare meiosis 2. Yes, we compare meiosis 2 with this mitosis. It is ditto like that, because n number of chromosome in the parent cell and you will get the n number of chromosome in the daughter cells. ठीक है बच्चे, that's the point. चलो, okay, ठीक है, that's the point, right? See, it's like, जैसे I usually give this example, मतलब interkinase is a very small gap. ठीक है, you know that usually in telophase what is going to happen? In telophase, the chromosome again they will decondense, again they will lose their individuality, again they will become tangled, right, like this. But again, it will not be completely decondensed. Matlab it's like that ki you were there in the market, you came home, you know after 30 minutes you again have to go to the market. ठीक है, so you are like, चल ठीक है यार 30 minutes के लिए let's take the rest. मतलब let's say you are wearing some trousers, you'll change, you'll wear some shorts or something like this, but you know that after 30 minutes again you have to go to the market. So you have that mindset, right? Back of the mind you have that thing, yaar I have to go to the market. You're not going to change your clothes completely, you're not going to come in that comfortable clothes of yours. You're like, ठीक है यार, again I have to go. Same is the case here. Now this cell, it has to divide, so the complete decondensation will not occur. Complete condensation of this chromatin material will not occur. Now let's move to the prophase, right? Let's move to the prophase 2. Now we have two daughter cell. Here again bache, the things will be same. Your centrioles will move to the opposite poles, they'll duplicate, they'll move to the opposite pole, right? Nuclear membrane will disintegrate. Plus here the chromosomes again they will compact. They are not totally decondensed, but still they will show the compaction. Now this germ cell is also having 4 chromosomes. This germ cell is also having 4 chromosomes. Okay. Now when you talk about the, right, so prophase mein same things will occur. Here you don't see any sub stage or something. Now let's talk about the metaphase 2. Now let's talk about the metaphase 2. Right, bache? Let's talk about the metaphase 2. In the metaphase 2, right? In the metaphase 2, see, because that cells are having 4 chromosomes, obviously they will arrange. So you will see bivalent in the metaphase 1 only. In the metaphase 2 it will be like this. Now again in the anaphase 2 also, just like mitosis, there is the splitting of centromere, there is the separation of sister chromatids. Are you getting my point? There will be the splitting of centromere, there will be the separation of sister chromatids. Are you getting my point? There will be the splitting, right? There will be the splitting of centromere and there will be the separation of sister chromatids. Are you getting my point? And again in the telophase. So now see what we are doing here. Here the chromosome number is 4 only, but the DNA content is 2C. Ultimately in the telophase, that will be the case, no? That will be the case, na? Your chromosome number is also half. But we are also, right, we also want the DNA content to be C, right? So now just imagine this is a sperm, right, having 4 chromosome and C is the DNA content. This is the ovum having 4 chromosome and C is the DNA content. So zygote mein you will see 2C and 4 4 8. 8 chromosomes will be there, and that is what we want. This is the initial amount there, right? This is the initial amount there. Understood? Right? Done, bache? Yes? Done? Okay? Done? So that is what we have. That is what we have at the end. So this is mitosis, this is meiosis, right? I hope it is clear. I told you what are the differences in the metaphases. I told you what kind of changes are going to be there. So you guys can see this picture also. Just focus here. Metaphase 1 mein there will be the bivalent at the equator, right? There will be the bivalent at the equator. Telophase mein, matlab obviously anaphase mein there is the separation of bivalent. No sister chromatid will get separated in anaphase 1. Then telophase, metaphase 2 mein individually the single chromosome. Here you are not going to see the pair, single chromosome. Then their sister chromatids will separate. Their sister chromatid will separate. Right, bache? So this is all about this chapter. Okay, this is all about this chapter. Diagrams, ye dekho. Metaphase. Metaphase 1, pair. Right, anaphase mein that pair will separate. Finally, telophase. Right, then again, prophase 2. Then in metaphase 2, you can just see the number of chromosomes of half. Then in anaphase again, anaphase 2 again, splitting of centromere and the separation of sister chromatid will be there. That's it. That's it. Okay? So these diagrams will help you to understand the topic in a better way. So this is all about this topic. Now I will discuss some checkpoints, bache. Then obviously class will be over. Right? Let's talk about the checkpoints. Yes, let's talk about the checkpoints. Okay? Okay, checkpoints are important. It is not there in NCERT, but still, bache, it is important. Right, bache? So, when you talk about the checkpoints, you have, yes, you have G1 to S checkpoint, G1 to S checkpoint, right? For the progression of this cell from G1 to S, you have G2 to M checkpoint also, right, bache? Then we also talk about the APC, anaphase promoting complex. Jaise dekho, about this I already told you, right, that when there will be a go ahead signal, there is in the animal cell there is a restriction point. So once a cell has crossed it, it will enter in S phase, it will continue the cell cycle. So when it is the G1 to S transition, yeah, progression of cell from G1 to S phase, so obviously what should be there? The cell size should be proper, right? The proper growth factors should be there, right? The DNA should not be damaged, right? The DNA should not be damaged, right, bache? So these are the points. If everything is fine, right? If the cell size, if growth factors, if DNA is not damaged, if proper nutrients are there, cell will enter in the next phase, right, bache? Here also G2 to M. So if DNA replication is proper, if there is no problem in that, right? DNA replication, right? If there is no DNA damage, if the cell size and all the nutrients are available, it will go for this. And now, bache, when we move from metaphase to anaphase, there is anaphase promoting complex, right? There is anaphase promoting complex. So these are some checkpoints. They'll keep a check, right? Why? Because the cell division should be proper. Just even when we talk about the DNA replication, you know that mutations are allowed but at a very slower rate, right? Right, they are allowed but at a very slower rate, right, bache? So if let's say DNA is too much damage, obviously we cannot let that cells to grow, right? It can result in the cancer. It can result in the cancerous cells, right? So we have to take care of that. Okay, we have to take care of that. So that type of checkpoints are there which will take care of the things. So when we talk about the checkpoints, we'll talk about some proteins, this is cyclins, and some CDKs, cyclin dependent kinase. Cyclin dependent kinase. So there are different types, cyclin A, B, a lot of types of cyclins are there. So they are, you know, they'll periodically form, break. They are acting like a checkpoint. Okay? They are acting like a checkpoint. So that's it. That's all, right, about the checkpoints. Plus colchicine. Do you know what colchicine is? It is also known as mitotic poison. Mitotic poison. It is used even, right, to study the cancerous cells. So colchicine. Do you know anything about colchicine? Yes. Do you know anything about colchicine? If it is a yes, very good. If not, then obviously you have to read about it. Okay. Right? So if we have to study the cellular genetics, we need this. So colchicine inhibit the polymerization of, it inhibit the polymerization of tubulin. It will not allow the tubulin to polymerize. Okay? It will not allow the tubulin to polymerize. Okay? And obviously microtubules will not form, spindle apparatus will not form. So obviously how that chromosomes will get separated. So in cellular genetic study, it is used, right? Scientists, they introduce it, right? They don't want that chromosome to get separated. They want to study them. Okay, they want to study them. So yes, even you need to know that, right? So number of, just sometimes they ask, number of generations required. This is a very repeated, this is a repeated question. Number of generations required to produce. Yes, number of generations required to produce X number of cells. So the formula is 2n there, right? The formula is 2n there, whereas n is the number of generations, right? N is the number of generations. So if you have number of generations, right, so you can just calculate the cells, and obviously you can use this formula. Okay? Okay? Okay. Hanji, any doubt? Any doubt? So this is all about this chapter. If there is any doubt, please do let me know. Right? Do let me know. Baki, just read about it, solve the question on the basis of that, and trust me, this chapter is not very difficult. It is easy. If concepts are clear, right, then everything is easy. So prophase 1 will take time, then metaphase 1 may, yes, then in the metaphase 1 you have to be clear, it is the bivalent that will be present here. Bas, that's it, that's all. Okay? So I will share the PDF in our Telegram group, right? The link for the Telegram group is also there in the description box. You can go there. And guys, please, without wasting your time, be the part of the batch, because batches are really going to be amazing, right? Bache, even the all-star fast track crash courses batch is also there. You can join it, where, you know, many teachers are teaching the chapters, right? So you will get a chance to complete your syllabus from all your favorite teachers. Okay? Right? Chalo. So do let me know in the comment section you like the session or not, and do mention the homework there. So thank you. We have the next class on Monday, and that is plant kingdom. So let's finish plant kingdom, then animal kingdom, biological classification, biomolecules. So we have to finish the syllabus of class 11th in the November only. Okay? Chalo. Bye-bye. Take care.